Welcome, everyone. It's nice to see so many friends interested in this very easy topic, right? Normal pressure hydrocephalus. I also want to say hi to all the people that are joining us virtually. I understand there were at least 150 people registered for this little symposium we put together. So clearly showing that there's an interest in this controversial yet very interesting, in my opinion, topic. So welcome everyone. And first of all, I want to thank you all and the speakers for taking the time to discuss their view on NPH. I'm Alfonso Fasano. I'm a neurologist here. I'm going to be chairing this event. The other co-chair, as you can tell, is not Gelare anymore. Gelare unfortunately had an issue with her plane. I mean, she's fine. The plane was delayed and she's trying to be here as soon as she can. I don't know what time she will be able to arrive. The good news is that she's landing at the city airport, so we will see. Because she needs to have an helicopter at some point. Anyways, we're going to be covering a number of topics, and I now leave the mic to Gabor Kovac, who needs no introduction, and thankfully agreed to co-chair the session with me. Please, Gabor.
So good afternoon. My name is Gabor Kovac. So no more pressure, but normal pressure hydrocephalus. And it is a pleasure that we have so nice speakers. We will have speakers from Uppsala, Sweden, from Cincinnati, and modestly from Toronto, myself, and of course, our famous normal pressure hydrocephalus researcher and clinician, Professor Alfonso Fasano, who I kindly asked to open the session.
Thank you. So I'll be just introducing the topic and setting the stage for the issue of this term, normal pressure hydrocephalus, and particularly idiopathic. As you know, the title of the event is what is idiopathic normal pressure hydrocephalus? So these are my disclosures, and I highlighted here the ones relevant to this topic. Even though they are not really relevant to what I'm going to say. So this is the Western. This is the topic of our event today. And I want to start with a little bit of history. And that's actually quite interesting. This is something that Charles Taylor sent me when he heard about the symposium. And he said, oh, interesting. I started to look into this in 1967. And I looked at the publication. Actually, was a resident back then. And so it was 2 years after NPH was described, as we know, in the famous paper in New England. So a few words about idiopathic. In medicine, we don't like it. Anytime we hear idiopathic, it's basically testifying that we don't know what we're talking about. And in this recent viewpoint we put together, and Alberto is one of the authors, and also David Tangway, we concluded saying that hopefully in the future we won't be dealing with idiopathic. Like I said, I'm going to just set the stage, and in doing so, I just want to give you 4 different scenarios of why it is difficult for us to deal with the complexity of this condition, especially when it comes to pathology, as you'll hear later. Because often you'll see that people are diagnosed with multiple disorders, or even a pathology, they're found to have different disorders. And this can be just coincidental. You know, bad luck exists. These are old patients, and often they have multiple problems. But there is another situation where NPH itself, problem with CSF circulation in the central nervous system, can alter the clearance of proteins if we believe that proteins are toxin. And I know that most of you believe so, but there's one person who's pretty vocal about it, and we'll probably hear from him. And that is Alberto, of course. But I guess we're now emphasizing the role of the lymphatic system. It is a recently discovered system. I won't go into the details, but it's probably a very important key player in many diseases. And importantly, also certain proteins like aquaporin 4 are relevant to the topic of absorption. And what's good about this system is that we can now see it. There are MRI techniques that give us the opportunity to see the flow in the brain. And important, when you see what happens in normal pressure hydrocephalus, you see that the CSF doesn't really move. You see retrograde flux back in the ventricles. And so that led to the hypothesis that some of these toxins can stay and damage the brain. And this is a recent paper when they looked at the toxic form of amyloid, the beta oligomer, which were found to be increased in NPH. And interestingly, these authors, they looked also at the effects of shunt surgery, and they determined that this the level of oligomers goes down after shunt surgery in people who improve. Likewise, there have been interested in dopaminergic effects of NPH. If you were to do that scan, which we don't do often in Canada, thank God, but if you do more, you will see that a lot of patients actually have a normal dopaminergic transmission. And actually in this paper, we found that 62% of patients had a normal DAT scan. Does it mean that they have Parkinson's disease? No, they don't, for a number of reasons. First of all, it's a mainly caudate involvement and is very symmetrical, unlike Parkinson's disease. But we know that over time, especially if you don't shunt them, this worsens. But at the same time, if you shunt them, so this is the one worsening over time, if you shunt them, the DAT scan binding remains the same or actually even gets better. So 62% is the quote in this paper of people with a normal dopaminergic transmission. But when we did, and this is something I've done with Carmela Tartaglia and Gabor, of course, Gabor Kovac in his lab, when you do RT-QuIC looking for alpha-synuclein pathology in these patients, you only find it in 14%. So there is a quote of them with a synucleopathy, and they don't really differ too much from the negative ones, although they respond more often to levodopa. And as you can imagine, in the long term, they don't do as well. And this has been replicated by another study, which basically came up with the same number, more or less 20% of people with pathology, synuclein pathology, as opposed to the 62 that we can see with imaging. Then there are other people arguing that this can be that the ventriculomegaly can be just a presentation of neurodegeneration. So this has been called hydrocephalic presentation of neurodegenerative disease. And some of the papers that is the most quoted in this field comes from, is this one. This comes from the Mayo Clinic. But I also want to start with saying that these are 41 patients collected over decades. This tells you that there was a bias in selecting patients, I think. This might come from a center where NPH doesn't exist. We see 41 patients in a year, 41 new patients in a year. But anyways, this is a pathology study, and they found that some of them, a very small proportion of them actually went for a ventroperitoneal shunt. Some improved, and interestingly, also the ones who improved were found to have another alternative diagnosis at pathology. But that doesn't necessarily contrast with having NPH. Often we see, and it's published many times, tauopathy with NPH. And one of the first times I spoke with Gabor, it was after his presentation when he showed data on certain types of tauopathy where, for reasons unknown, there was an increase of ventricular size that wasn't only explained by atrophy. And we should remember that here we're dealing with a disorder of the cilia. And cilia are made of tubulin, and one of the proteins that has to do with tubulin is tau. Clearly, the link is not established yet, but more and more new papers are coming up looking at certain genes and proteins involved in the physiology of tau and the tubulin process, which are part of cilia. So a possible connection might be there. But back to Alzheimer's disease, there's a recent paper where they argue that there is an hydrocephalic variant of Alzheimer's disease. I don't know if there is anything in there, but I find interesting that in this hypothesis, they basically came up with two alternative situations, an NPH first hypothesis versus an Alzheimer's first hypothesis, which I think will generate some discussion. And finally, what if we deal with something that is upfront, something that causes ventriculomegaly and from there neurodegeneration? And I think here the link comes from familial studies of NPH. So there are families of NPH, and these are the genes identified so far. And with the exception of C9orf72, all of them are related to cilia physiology or structure. And this is one of the first publications where not just the animal model developed hydrocephalus, but also in terms of structural abnormalities with electronic microscope, the cilia of these affected members were non-functioning. So the hypothesis is that the cilia don't work on the flow very well, so they don't regulate in a organized and patterned way the flow of CSF, and over the years you develop ventriculomegaly. And the cilium is a very complex structure. I don't think I need to tell you, but in case you wonder, and if you want to test the genes for ciliopathy, this is the panel at this moment. And interestingly, the panel does include the genes involved in familial hydrocephalus. And finally, taxanes, these are chemotherapeutic, they can induce NPH. And the mechanism is the same. It's known that they have an effect on tubulin and microtubules. That's why people often have neuropathy. And for some reason, some people also develop normal pressure hydrocephalus. This particular subject here got even treated. And now, since I started with Charles Taylor, and we all know he's interested in trauma, I also want to make the point that there is a blurry line between what we call idiopathic and secondary forms of normal pressure hydrocephalus. Think about post-traumatic, post-meningitic, or any other post-subaortic hemorrhage type of NPH. And a possible link comes here. This is something we're working on right now. We looked at the history of any traumatic brain injury 5 years at least before the diagnosis, and we found a high prevalence of such history with a very documented way to analyze the data with 2 scales. We used 2 different scales to be redundant, and we found that up to 80% almost of NPH patients labeled as idiopathic have a history of traumatic brain injury. So in short, this is what I think we're dealing with. I think we are certain individuals who are predisposed based on genetics, and these genes probably have to do with cilia function. Cilia are functioning, otherwise these people will have respiratory issues, they will not be fertile, but they're probably not functioning well enough. And with additional effects of environment, including drugs, this can get worse. And CSF dynamic starts to be impaired. This is when ventriculomegaly starts to develop. And often, I would say almost all the time, you talk to these patients, they have sleep apneas, which is seen as an important risk factor, as well as other vascular risk factors. So these are probably contributing to the development of this problem. And then if you wait too long, the brain gets damaged. Maybe there is pathological accumulation. And this is where you're basically dealing with 2 situations: ventriculomegaly first, with no signs, then a proper normal pressure hydrocephalus that can be treated, and then neurodegeneration. And obviously, if you wait too long, and this has been shown by studies, to treat them, there is no good outcome. How do I know that we can have hydrocephalic evidence without signs? Well, there are enough studies now looking at this. And the percentage of people reported are almost the same, regardless of the country. Japanese people have worked a lot on this. This is a cohort followed for 16 years. They looked at people with ventriculomegaly, and they saw what happened to them. And a part of them developed NPH, especially the ones starting with DASH. And finally, like I said, I just wanted to set the stage to explain why we felt that it was important to discuss what idiopathic really means in this context. I just want to say that in case you're interested, within the Movement Disorder Society, we put together a study group, because for the longest time, neurologists and movement disorder neurologists weren't interested enough in this topic. So if you're interested, just let me know. And finally, I want to thank you all, the fellows, the support we get from UHN, and the people involved in the program that we have here. And thank you all for their attention.
Thank you, Alfonso. Do you expect questions now, or we should do it?
We're going to have a panel discussion,
okay.
And we are just 1 minute late.
Yeah.
I don't think we can address, because it was just to introduce the topic. In case someone has some burning question, I'm happy to answer, or we can move on to the next speaker.
Thank you very much again for the wonderful introduction. And I would like to kindly ask Silvia Libac. She's from Uppsala University, Sweden, and she has a great experience in studying normal pressure hydrocephalus from the neuropathological side, due to a lot of biopsies carried out during the shunt surgeries. So we hope that we get her insight.
So Thank you very much for the opportunity to show our experience of this subject. My name is Silvia Libard, and I'm a neuropathologist at Uppsala University Hospital in Sweden. And as the neuropathological community have different specialties, both medical and scientific. So I have a little background slide. So I'm actually a general pathologist. I got licensed in 2013, but started with my neuropathology career already 2009 when Professor Alafuzov moved to Uppsala. I defended my thesis in the topic of normal pressure hydrocephalus in 2021. I have some examples with me, as our books are quite special, so if someone wants to have one, you're welcome. But now to the main topic of the day. So it all started in Finland. Finland had centralization of healthcare during the 1990s. And Kuopio University of Eastern Finland, with a population covering about 800,000 people, was responsible for epilepsy surgeries, NPH sampling, and dementia care. And there were two main people to develop this technique, and it was a neurologist, Paavo Reikkinen, and neurosurgeon Matti Vappahalahti that studies this patient group and decided after a meeting to biopsy those patients, as most of the subject displayed mild cognitive impairment and progressed into dementia. So they started with that in 1991, and up to 2016, they had about 446 samples. And as the biopsies are diagnostic, when A-beta was seen in the biopsies, the patient was referred for neurology department for further investigation. And in Uppsala University Hospital is first local hospital for 400,000 citizens, but it also covers subspecialized care for the region of Middle Sweden for about 1.2 million people. And when Irina came to Uppsala, she invited Professor Leinonen from Finland to Uppsala, that introduced the biopsy approach even at our hospital. And we started to diagnose those biopsies. And since 2010, we have around 600 biopsies in our biobank. And this is how it's done at our hospital. The biopsy is taken from the area of the shunt tunnel in the superior or middle frontal gyri, as you see there on the picture. And it's a needle biopsy that is approximately 2 millimeters wide and 1 centimeter long. We studied the tissue damage around this biopsy taking, which has been quite controversial. And we see the shunt tunnel that is 2 millimeters wide with reactive changes within an area of 4 to 6 millimeters with astrogliosis, some scattered lymphocytes, and foamy macrophages. And the tissue damage is along the whole shunt tunnel, actually, as the biopsy is only taken in the apical part. So the tissue damage is made mainly by the shunt itself. So the biopsies have been taken since 1990 in some centers in the world, but up to date, no neuropathological consensus criteria for NPH has been established. Post-mortem investigations are seldom performed. There are very few reports about post-mortem findings in those patients. The biopsy approach varies between different centers. We know that in Norway, the biopsy is taken during intracranial pressure monitoring, for example. And in Finland, nowadays, I think they take a little larger tissue samples than at our hospital. And what we see in those biopsies is that substantial number of subjects display Alzheimer's disease neuropathological change, so A-beta and HB-tau in the biopsies. And when it's present, it is associated with the worse shunt response. And many of those subjects progress into dementia of AD type. There are only a few cases with TDP-43 pathology reported, one from Finland, and we had one case with only one neuron containing TDP-43 pathology. And alpha-synuclein, the clinician often asks us about synucleinopathy in those biopsies, but we didn't have any positive case, as this is in the synucleinopathy. This is the last place to find the pathology. So I think, and I didn't find any neuropathologic publications regarding alpha-synuclein pathology in those regions. So, and as you know, Alzheimer's disease neuropathological change is beta amyloid pathology, and we see it as cerebral amyloid angiopathy, but very sparse in those biopsies, as meningeal coverings, which is the primary site for this pathology, is often lacking in our biopsy samples. And then we see those diffuse senile plaques and dense A-beta aggregates. And beta amyloid pathology progressed through predefined neuroanatomical regions, first affecting cortical regions, lately spreading downwards into limbic structures, central gray matter, mesencephalon, and cerebellum. So when seen in the frontal cortical biopsies, we see the pathology quite early, often in asymptomatic patients, maybe. And hypophosphorylated tau pathology, we see often as neuritic threats, and sometimes in our biopsies only as a few granuli. We also see neurofibrillary tangles and seldom neuritic plaques. And also this pathology is progressing through predefined regions according to Bragg stages, stage 1 and 2, only seen in anterenal regions, 3 and 4 in limbic regions, and 4 and 5 and 6 in neocortical areas. So when seen in the biopsies, even if very sparse, it suggests that the pathology is below, is over low. So it's at least intermediate. And we wanted to study this, and between 2010 and 2016, we collected 364 biopsies, and we wanted to study the ADNC and neuronal population in those biopsies. We chose an age rate of the patient where we suspected that we would see some pathology, and in the final study, 95 subjects were included. We assessed the pathology with morphometrical method, as you see here. And what we could see was that 63% of the subjects displayed A-beta pathology, and it ranged in extent from 1 to 27%, as you see at the upper row in the figure. H-beta pathology was seen in 61% of the cases, and it varies from 0.02% to 19%. And the example of 0.05 is on the lower line of the figure. What is worth to say is that a majority of the subjects displayed very low H-beta pathology, as it was below 1%. And concomitant pathology was seen in about 50% of the cases. We noticed that female displayed more alteration than men. For H-beta was significant and borderline significant for A-beta, which is in line with what is seen in postmortem status of AD patients. The two ADNC markers correlated with each other and with age, but what was quite surprising was also that the age correlated with one of the neuronal markers, and as we don't suspect that there is new formation of neurons, that is a sign of atrophy, and we tested it by looking at synaptic marker and synaptic density, and actually in cases where we could see more neurons, we saw synaptic loss. So this neuronal preservation and synaptic loss, together with this very low level of H-beta, is in line with what you see in early degenerative process. In parallel to the neuroanatomical progression of A-beta pathology, also you can also see some biochemical changes within the aggregates. Some of them is pyroglutamylation and phosphorylation of the protein, which is associated with increased aggregation, neurotoxicity, and cognitive impairment. And in 2014, a group proposed that this happens in a stepwise manner, that at the beginning you have where you have A-beta pathology without any alterations. At stage 2, you can detect pyroglutamylated A-beta variants within the plaques, and at stage 3, when the subjects have symptomatic dementia, also the phosphorylated variants are detectable. And we tested it on a NPH cohort of 127 subjects. We looked at 5 different A-beta variants. 6F3D and 48 are the most used clones. We had an unmodified variant without any biochemical alterations, phosphorylated and pyroglutamylated variant. And of course, all of them increased with age and correlated with HB-tau pathology. What was quite surprising was that in the 2 oldest age group, we saw a slight decrease in the expression, but only in females, which actually was in line with that most of the females probably are demented when they are so old and don't come to the biopsy. The level of pyroglutamylated A-beta was higher than phosphorylated in all age groups, suggesting that the formation of this variant precedes the variant of phosphorylated type. And this is in line with what's seen in AD. And some of these variants have been seen intracellularly in neurons. So we wanted to study this. And for that, we could sample 12 biopsies for electron microscopy. To be included, they of course had to display A-beta pathology and have gray matter areas in the electron microscopy sample. Actually, we could confirm extracellular localization of the A-beta aggregates in all methods we used, and it was light microscopy, semi-thin section in light microscopy, and electron microscopy. We could detect 3 different markers with this EM method. Unfortunately, this unmodified and phosphorylated type was not detected. The aggregates contained numerous particles, which seems that the method worked, and were often located nearby dystrophic neurites and swollen destroyed cells, but you couldn't define any intracellular expression of these proteins. We also looked at APP, and the expression was very sparse, as in both in intra-extracellular compartment in line with the location of this protein, as it's seen on the cell membranes and membranes of organelles. And this we took as a control to see where the amyloid species are located. We also had possibility to study different neuropathological processes in subjects diagnosed with both NPH and AD with 21 months interval. And the patient was a 63 years old female that debuted with cognitive impairment and memory loss. And after 2 years was diagnosed with AD at her local hospital. When she performed 25 mini mental state examination, but her MRI corresponded to NPH. She progressed really quickly in both motorical and cognitive symptoms. She had to move into nursing home just 1 year after her diagnosis. Her motorical function was worse and worse, so she was referred to a neurology department of Uppsala University Hospital. And when she arrived there, she could barely walk and not participate in the cognitive testing. She had pathologic cerebrospinal fluid parameters and liquid dynamic tests. And due to her quite young age and severe motorical function, they decided to offer her and her family this shunt, and she was biopsized by then. At our hospital, you have usual 3 months and 1 year follow-up, and she improved by those follow-ups. On the last one, she could walk by herself, and also she was talkative. She couldn't perform in the cognitive testing, but she answered some questions. She passed away at her local hospital 21 months after the biopsy, and autopsy revealed pulmonary embolism as a cause of death. and we got her brain for the neuropathological analysis. And the diagnostic biopsy displayed ADNC, and the post-mortem examination of the brain verified Alzheimer with Braak stage 5 and Tal stage 3. She had additionally the CAA type 2, ARTAG, and TDP43 pathology corresponding with late stage 2. So we could study different markers and the changes within the tissue during this 21 months. And what we saw was that the level of H tau pathology increased at the end stage of the disease. A beta was quite stable. A beta 40 increased, but this was due to the cerebral amyloid angiopathy that we didn't see in the biopsy, but was present in the postmortem brain. We studied 4 different neuronal markers, and all decreased over time. What was interesting that we also studied some inflammatory markers, and GFAP was quite stable, but the microglia markers decreased over time. So what we could see was the A-beta pathology was stable. We saw signs of progressive neurodegeneration with increasing H tau pathology and loss of synaptic and neuronal markers. Gliosis was stable, but microgliosis decreased. So postmortem examination from subjects with NPH should be performed because it can show us more pathology than we ever can see in those biopsies. We are also interested in risk factors for AD and other dementias. One of them is diabetes mellitus. And there was several epidemiological studies that show association between AD and type 2 diabetes mellitus, but we couldn't confirm it with neuropathological studies. We found several publications that showed the expression of islet amyloid polypeptide, also called amylin, in the brain. And this is a pancreatic B-cell protein that in pathologic conditions is the main constituent of pancreatic amyloid. So we studied this in those biopsies, but also in postmortem brain tissue from in a tissue microarray from postmortem brain tissue, as most of the previous study were performed on postmortem cases. With 2 different antibodies, a monoclonal one and polyclonal, both specific for the beta cells in the pancreas. So in the TMAs with postmortem brain tissue and the polyclonal antibody, we could see expression in line with previous studies. That was in some cases intracellular, plaque-like or vascular, but we couldn't confirm it with the monoclonal antibody used. And on the NPH cohort, we only used the monoclonal antibody due to a shortage of tissue, and we couldn't confirm the finding. There was a recent study in 2021 by recent colleagues that tested 8 different commercial amylin antibodies, among them the one mostly used. And it showed that amylin shares 40 to 50% of amino acid sequence with a neuronal protein widely expressed in the brain. So this expression with polyclonal antibody can be caused by cross-reaction of the proteins. So in our studies, we could see no association between type 2 diabetes mellitus and AD when assessing the neuropathological outcomes. So the summary of what we see in those biopsies is that majority of subjects display ADNC. The level of H tau pathology is low. There is exceptional neuronal preservation and synaptic loss. Both pyroglutaminated and phosphorylated variants are detected, and it seems that pyroglutaminated variants precede the formation of phosphorylated. The A-beta pathology is seen in extracellular compartment. And at the end stage of the disease, there's a progressive neurodegeneration. A-beta pathology is stable and neuroinflammatory markers decrease. And we could not see any association between ADNC and type 2 diabetes mellitus in NPH subjects. So my conclusion is that substantial number of subjects with NPH also suffers from AD. The diagnostic biopsies are of great value as the information achieved can benefit the patient's future care, while the tissue damage is sparse. And a post-mortem examination should be obligatory, as we only see those changes in 50% of cases, and we don't actually know what happens in the other 50. If they have A beta pathology, but we don't see them in the biopsy, if they're a vascular disease, or if it's other changes. So I think post-mortem examination should be obligatory in this patient category. I want to thank my co-workers and founders, and thank you.
Thank you very much, Sylvia, for the comprehensive and interesting overview. Anybody questions from the audience?
Maybe I can start with one question. Because, so first of all, I'm a big fan of biopsies in procedure. Also, when we do DBS, I think we should do biopsies more often. And thankfully, now that Gabor is here, I found a buddy that agrees. But you make the point that should be done because it helps the clinical care of patients. But I think it helps the understanding. But does it change the clinical care in your experience?
Well, at our hospital, when A beta pathology is seen in a biopsy, the patients are recommended to a mammo clinic. The healthcare system looks a little bit different because they only biopsized at our department, and the investigation and follow-up is made of the neurology department. But then they are either referred back to the primary care units, or when you find this ADNC in the biopsies, they are referred to memory clinics when they can be investigating for demented illness.
But they will still receive the shunt.
Yeah, they will still receive, yes, because the biopsy is taken during the shunt insertion.
So it's actually what happens in terms of follow-up to the patient that makes the difference. Interesting. So, and if not, which is a minority, they will only be followed by neurosurgeon.
Yeah, they have 1 year follow-up, and after this 1 year, they are referred back to the GP.
Community.
Yeah.
Interesting. Okay.
I would like to ask that I see that you have a long experience, like many years. Do you have any feedback after the follow-up of these patients that you biopsied, let's say in the beginning of your study? Do you ever receive any feedback? What happened with those patients? How long they lived? What?
No, and this is
whether they really proceeded to a whatever neurodegenerative disease. Or...
She separately based on those who had A beta biopsy and those who didn't have. Sorry, yeah. So, but this is in the Finnish material. So there was a neurologist who went through all the cases, and principally all those who had beta, they evolved into Alzheimer's disease. Whereas regarding those who didn't, there was some who got actually diagnosis of vascular dementia. So there were some people who reasoned that maybe there is a vascular dementia in the background. Another were quite stationary with shunt. Regarding Sweden, we have a couple of cases where we had tau pathology and no A beta. And we really wanted the clinician to look at them in the case that have they developed to a PSP or CBD or something else, but that is not done.
No, and the problem is the system, because we lose the patients when they are referred back to the community.
Yeah, it's the organizational system. So it's Sweden is characteristic in that all the medical healthcare is regionally oriented, and at the same time special clinics, they serve several different regions. So if we shunt a patient in our region and then the patient is transported to another region, we lost it. For example, this patient that Sylvia had the possibility to look at the postmortem brain and the biopsy, we got the brain because the husband had required that when the wife dies, the brain should be sent to us. Otherwise, we would never have got it.
Yeah.
So there are structural problems.
And if I might have one more question regarding neuroimaging. So in the biopsy cohort where you find beta amyloid, where you don't find, do you have anybody who looked at MRI variables like the proportion of the different parts of the ventricles? So, or even in follow-up. So is there any hint from the neuroradiology that this is an Alzheimer type or whatever
Not yet,
type?
but I think we will do this in the future.
Okay.
There is Charles Taylor. Dr. Taylor was raised his hand. I don't know if you can unmute.
Yes, can you hear me?
Okay. Yes, we can hear you.
Oh.
Unfortunately, I'm looking at the screen, but people can't. Yeah, but they can hear you. Don't worry. Go ahead.
Oh, perfect. Thank you, Alfonso, for organizing this. It's terrific so far. Is there any indication from the pathology that Dr. Libard studied so carefully as to the cause of the hydrocephalus part of this condition?
No, not in the studies I made so far. We mostly see the ADNC, and it was what I have focused on.
Like there's no meningeal component or?
No, as I said, there are very few. I see the meningeal coverings in about 5 to 8% of the cases. So a very few cases that still have the meningeal coverings in the biopsies. So.
Thank you.
Yeah, there's one more comment from the audience. Yeah, and
This is not a question. I just want to comment this because what we have been struggling with, really struggling with, is that we don't get these cases postmortem. So principally, we have tried, actually. We go to the morgue and we see to that those that come in, we also get for investigation. But it's seldom that the postmortem investigation is asked for. And that at least makes me a little confused, because if you have a patient and you don't even know what he suffers or she suffers from, and then this subject dies, that you don't try to get that information. It's that I think it's very, very important to try to get more of those cases.
Yes, and I mean, they have undergone a surgical intervention, so it's why even more should lead to a postmortem investigation.
Yeah, got the message. More biopsies, more brain autopsies.
Yeah.
Please.
Yeah, thanks for your talk. We've been doing biopsies for a long time as well. But what are the criteria for the diagnosis, and what are the criteria for shunting that you have?
I don't know. The criteria for shunting is for our clinical colleagues. We are not involved in this. We diagnose the tissue, and I just describe the level of pathology. So I write that it's few or few threads of the tau, and I see A beta pathology. Most of the studies that we have described this pathology and progression into AD only describe that the pathology is present or not. So we don't stage it.
Yeah, but this is an important point, because for example, here we are very reluctant in doing anything in people presenting with cognitive disorders as first problem, or with a remarkable progression of that particular domain with amnestic features. We really consider NPH as a motor disorder. And I'm biased being a movement disorder neurologist, but the best outcomes we have so far are in people with gait disorders, freezing of gait after surgery. Mobility improves. And I bet that if you were to look at the biopsy of these patients, I'm sure we'll see Alzheimer's pathology in many of our patients, but not so often in this particular type of patients. This is probably where this question comes from. Yeah, I guess it requires more collaboration also in understanding how these patients presented. We all agree that the evolution is different and they tend to do worse. But the question is, can we use this to look back and maybe be better diagnosing co-pathology in these patients?
Maybe one more question. That do you have any observation or data on the response to the shunt in these patients, like A beta versus no A beta?
This is again from Sweden, because I had more summary experience there. And principally those who didn't have beta, they responded better regarding their gait problems, etc. Whereas, and I think from Uppsala also there is a study where the response was not as optimal if you had the beta. And just to respond to this question regarding when the patient is shunted, is usually because when they have lots of gait problems and urinary incontinence. However, some of them have always might have slight cognitive impairment, and that does not exclude them directly, because you want to treat the gait and the incontinence.
Yeah, those are the ones responding. So any more questions? Also want to remind you all, also people from home, that if you want to ask questions, feel free to use the chat box. We might not necessarily be paying attention to features like if you raise your hand on the virtual platform, but chat box is easy for us to keep track of. So don't be shy. Feel free to type in. Okay, if there are no other questions, I think we can move on to the next speaker, who is at this point well known to you all, Dr. Gabor Kovac, who is now breaking the microphone. Okay, very good. Gabor is a colleague, but also dear friend. We're all very happy that he decided to come to Toronto for the weather and the food, as I always say. No, but jokes aside, as I said before, it was a great addition also because we started a number of collaborations, and he started a number of other collaborations with many people because there's a lot of things to work on and to do. And he's going to tell us about this topic that I mentioned before. So ventricular enlargement in people with neurodegenerative disease, where it's coming from. Is it just atrophy or is it something else? Please, Gabor.
Dear Alfonso, thank you very much for the kind introduction, and thank you for the opportunity to present here. So let me start with a personal observation. So roughly 4,000 days ago, which is about 11 years, I received a brain from a neurosurgical institute. And the question was that the patient has a very classical normal pressure hydrocephalus, but the shunt surgery is just not responding and deteriorating very rapidly. So on the neuropathological examination, I found a Lewy body disease. And the unexpected striking observation for me was that the aqueduct of the brainstem was very narrow, and the subependymal area kind of bolted into the aqueduct, and it was full of synuclein. So at that time, I thought that maybe this subependymal area and the innervation of the aqueduct might be affected in neurodegenerative diseases. So when we talked about this symposium with Alfonso, and one major reason was that Irina Alafuzov and Silvia Libat was visiting us, so we used the opportunity to organize this. So some months ago, I said, okay, I go back to this 11 years ago vision, and I try to do a study, and that is what I would like to present here today, which is not published yet. And with this study, I would like to kind of address the question that if our problem is that we cannot really know whether it is a neurodegenerative disease in the background of ventricular enlargement, or is it a disease without neurodegenerative disease? So that is what I would like to address. And I would like to go to my last slide first, after the conflict of interest, which is the acknowledgement slide, because first I would like to thank the colleagues who helped me in this, and then I will start the presentation. So I had the unique opportunity to collaborate with neuroradiology colleagues like Alexander Butte, Jürgen Germann, Shelley Forrest from our team, who helped with the neuropathology, and Mikhail Malik, a medical student who also helped with the neuroimaging data. So the topic and the collaboration we used, and this is just shows you also the opportunities that we have here in Toronto, is that the patients who underwent MRI examination during life and unfortunately then ended and deceased, and we had the neuropathology examination. So we had the opportunity to compare certain MRI variables with the neuropathological examination. And that is what I would like to present here. And actually, we ended up more than 30, 35 cases, which is a nice cohort. So that you understand what I really wanted, or we really wanted to examine here, I would like to provide a few definitions and also about the ventricle enlargement, what is known. And then I'll show you the study that I'm talking about. So to understand the components of neurodegenerative diseases and the aspects leading to ventricle enlargement, we have to understand that neurodegenerative diseases are characterized by regionally different amount of nerve cell loss. And based on the last 20, 25 years molecular biological studies, it seems that at least in the postmortem examinations, we see altered proteins in these brains, and these can be extracellular or intracellular, and importantly in different cell types, and not only in nerve cells, but also in the supporting tissue. And as you have heard, ventricle enlargement might have some connection with this supporting tissue. So when you see a patient with a clinical phenotype, you are thinking about vulnerability patterns that include based on a genetic background, but the anatomical, the cellular, and the protein alterations. And this is associated with the alterations and dysfunction of glial networks. So if we want to talk about ventricle enlargement, we have to understand which of these components go wrong and lead to the ventricle enlargement. So currently, this is how we classify neurodegenerative diseases, always starting at the clinical level, but most importantly, at least for the ventricle enlargement, the anatomical predominance of neuronal degeneration is very important to know, because there is this old kind of terminology that the ventricles will enlarge on that horn of the ventricle where you have an atrophy or nerve cell loss. So if you look at the different types of neurodegenerative diseases with the different clinical phenotypes, with the red box, you see the highlighted anatomical regions. And this already shows that there might be differences in the components of the anatomical components of the ventricles that are in the ventricle enlargement. So perhaps looking at the different parts of the ventricles, or even the aqueduct, or the third ventricle and the fourth ventricle, we might have even hints for the background of or associated neuropathology. So let's see what is currently known. And I found this image, and as a fan of UFOs, I thought that this is a confirmation of that aliens came before the humans, but then a very realistic, rational neurosurgeon writes that this is a hydrocephalus case hunted by ostriches 10,000 years ago. So it seems that this hydrocephalus has a history, and obviously the neurosurgical history in the beginning of the 20th century focused on the hydrocephalus, which is associated by the blockage of the circulation associated with tumors or developmental disorders. And the concept of normal pressure hydrocephalus without this component only developed later. And as has been mentioned, there are several components of this pathogenesis. And this includes the ependymal lining, the brain vascular system, including the lymphatic system, the meninges, and also the choroid plexus. So if you think about neurodegenerative diseases, then actually some of these are easy to associate with neurodegenerative diseases, because as you know, the Alzheimer-related amyloid can be deposited also in vessels, can be also obstructing the interstitial fluid drainage, but less known, and this is what I would like to address in this study, there are protein deposits and pathological alterations which are associated with the ependymal lining, and even, and I will show you this, something is going on in the choroid plexus. So I will not really focus on literature review here, so rather focusing on my study, but I still picked a few literature data, such as supporting the idea what I mentioned, that, for example, the distribution of the ventricle enlargement might be helpful even to distinguish different neurodegenerative diseases, here shown by a difference in the posterior part in Lewy body disease versus Alzheimer's disease. Or there are studies which show that in Parkinson's disease, if you do longitudinal MRI studies, then there is a slowly progressive ventricle enlargement that can be associated with the progression of the pathology. So eventually this can be, the ventricle enlargement evaluation can even be a surrogate marker for the progression of pathology in Parkinson's disease. Then there are studies which show that the patterns of atrophy in major neurodegenerative conditions with movement disorders are different, and especially that whole ventricle system, ventricle enlargement, is more what characterizes PSP and multiple system atrophy in compared to other conditions. Finally, and as has been mentioned, normal pressure hydrocephalus can be missed or mistakenly diagnosed in PSP or PD patients. So this shows that there is a vague border between the neurodegenerative conditions and this kind of idiopathic, which we, in a way, don't really know whether it is idiopathic, because as Sylvia mentioned, we don't really have longitudinal neuropathology studies, whether these are always idiopathic with nothing in the brain and maybe ependymal disease, or mostly associated with some kind of neurodegenerative disease. So what did we do in this study? So we evaluated neuropathological variables in individuals where in vivo MRI data was available. So we ended up with a cohort of Alzheimer's disease, Lewy body cases, corticobasal, progressive supranuclear palsy, and MSA cases, and the age is shown here. So generally, the MSA patients were, of course, younger. The sex ratio was relatively similar. We evaluated the different pathological variables, such as the presence of argyrophilic grain disease, which is a limbic condition associated with tau. We evaluated TDP-43 pathology, which is again a limbic disease, medial temporal lobe disease, the presence of cerebral amyloid angiopathy, hippocampal sclerosis, which would again lead to associate with the inferior horn ventricle, or amygdala Lewy body pathology. But in addition to this, we performed a systematic study focusing on the aqueduct of the brainstem, because this was my original idea, how the subependymal area in the aqueduct in the brainstem functions. And the reason why I thought that this is important, because we have a kind of, the cerebrospinal fluid circulation has a kind of a pulsatile component, and I hypothesized that if the subependymal area in the brainstem has some pathological alterations, then maybe this pulsatile dynamic of the cerebrospinal fluid circulation is altered, leading to a kind of accumulation of or bad circulation, leading to ventricle enlargement, which could be similar to the classical obstructive part type of ventricle enlargement, which was classically described with, even in children with primary aqueduct stenosis, when they are born with aqueduct stenosis, and that leads to a classical hydrocephalus in the children or child. So obviously we don't see a full stenosis of the aqueduct, and I don't want to say that, but what I say is that maybe the aqueduct and this pulsation of the cerebrospinal fluid is not normal. And this kind of leads to the ventricle enlargement without any tumor or any other thing. So therefore, we focused on the subependymal area and evaluated the synuclein pathology, the tau pathology, and also because everybody's talking about inflammation in neurodegenerative diseases, so we also evaluated whether there is any inflammation in the choroid plexus, which produces the cerebrospinal fluid. And there are conditions which do have inflammation in choroid plexus, like inflammatory diseases, which then lead to, again, an altered cerebrospinal fluid production and leading to ventricle enlargement. So let's see what really happens in this cohort that we examined. And this is practically what I told you here, summarized in this slide. So just very briefly, this is the method that we use. We use a morphometry software called Halo, and the upper part of the slide of this slide shows the choroid plexus. So we annotate the area that we want to evaluate, and this is the aqueduct where we annotate the subependymal area, which is this very, very thin, very thin, I don't know if you see it on the slide, but this very thin area around the aqueduct, which has very, very small amount of cells. And this is what we see, that, for example, we counted the CD8 positive cytotoxic T cells, and the red dots are what the software detects as a cytotoxic T cell. And then in the area, it tells you how many cytotoxic T cell you have. And this is the evaluation of the tau pathology, and this is the subependymal area. So you see this very, very thin line here. So this is the subependymal area. This is the ependyma. And this is where we evaluated the presence of tau or synuclein pathology. And just imagine that these are very, very thin neurites coming from the upper part of the brainstem, like the tegmentum of the brainstem, reaching the ependyma. And actually, you see also pathological protein deposits in the ependyma. So that means that we thought that this might correlate with this. So this is in yellow. This is what the software detected as pathological color. So the brown is the pathological protein deposit, but then the software converts it to this yellow, and this is the subependymal area. So you see that there is protein deposition in this area, and this is the synuclein. So this is just the first overview that the Lewy body condition has much more synuclein in this subependymal area than the multiple system atrophy cases. And then let me show you a few other pathological variables before showing you the MRI data. So for example, the subependymal tau pathology is very high in Alzheimer's disease, of course in PSP and CBD, and much lower in the Lewy body conditions. And we evaluated the pons and the midbrain separately. And this is, for example, some representative slides, and we are searching for these little brown deposits here. This is the tau in PSP. So in the subependymal area, you do see a lot of these tiny little neuritic components. This is an Alzheimer's disease where you also see these neurites coming to the ependyma and accumulating in the subependymal region. Now in this slide, I would like to show you another pathology. This pathology is in the astrocytes. The astrocytes are the supporting tissue of our brain. And there is a type of pathology called aging-related tau astrogliopathy. And in this condition, which is mostly seen with aging, you see the accumulated phosphorylated tau, so the same kind of bad tau that you see in Alzheimer's disease, in astrocytes in the subpial area, in the subependymal area, in the perivascular astrocytes, in the white matter, or in gray matter astrocytes. So in our study, we evaluated whether there is subependymal age-related tau astrogliopathy in these cases and whether they correlate with any MRI variable. And we evaluated whether there is any subpial aging-related tau astrogliopathy, which is abbreviated as ARTAG, and I will use that term. Just referring to a study that we published in 2017, interestingly, in the aging brain, these are the locations where you can find this aging-related tau astrogliopathy. And as you see, this is along the ependyma, along the aqueduct, or in certain white matter areas or in certain subpial areas. But the amygdala, the basal part of the brain, is a very, very hot spot for this type of pathology. Now, these are further pathological variables. So we did find some more cytotoxic T cells in the choroid plexus in Lewy body disorders. And we also evaluated the postmortem evaluation of ventricle enlargement. Now, these are the neuroimaging variables that my colleagues who I mentioned in the acknowledgement slide collected. So these are the variables, these 6 variables. So I will show you images that they kindly provided. So this is the aspects that they measure: the width of the lateral ventricle, the intracranial width, the skull width, the ventral horn distance, the intercaudate distance, the third ventricle width. So this is a MRI image which was not fully optimal for the intracranial width, and therefore it is more difficult to examine, but still these data still entered our database. And this is the further aspect that I mentioned in the previous case. So let's go through the MRI variables, whether there is any relation to the pathological diagnosis, and then I will go into more details. So first, I would like to show you that the scan time before the death, we have this data, and obviously for correlation studies, we will have to correct our observations, because some patients, as you see, had maybe several years before they died, they had the MRI. So obviously we have to keep in mind when I'm doing a postmortem study. So we correct the neuropathological variables for this scan time before death. But generally, the cases did not have significant differences. Now, let's see the MRI variables. So the width of both lateral ventricles was the smallest or lowest in MSA or MSA cases, and this was significantly lower than in AD, and also the Lewy body case was lower. The intracranial width was the highest in the CBD cases. The distance between the ventral horns was the lowest in the MSA cases. The distance between the caudate nuclei was high in AD and also in Lewy body cases compared to the MSA cases. The skull width was again smallest in MSA cases. These MRI variables correlated with the age of the patient, so we have to correct our observations to the age of the patient also. Now let's look at the pathology variables. So the width of the lateral ventricles correlate with the brainstem, with the tau pathology in the subependymal area in the brainstem, and also with the aging-related tau astrogliopathy in the basal brain areas. Now, just to interpret what does this mean, if we have in the basal part of the brain this aging-related tau astrogliopathy, that reflects a barrier dysfunction. So based on mathematical models, there was a model which predicted that the distribution pattern of this aging-related tau astrogliopathy in subpial areas follow the cerebrospinal fluid circulation patterns, which means that if it starts and accumulates in the basal part of the brain, that would reflect a kind of a traffic jam in the cerebrospinal fluid circulation. So interestingly, the ventricle enlargement correlates with this, reflecting that this type of pathology supports this traffic jam concept for ventricle enlargement. Now, interestingly, the subependymal synuclein pathology correlates with the distance of the caudate nuclei, so that part of the ventricle. is affected more with the subependymal type of synuclein pathology. Finally, the third ventricle correlates with both the tau and the synuclein pathology in the brainstem subependymal area, which also is, I think, an interesting observation. And finally, the T lymphocytes, interestingly, correlate very strongly only in the Lewy body diseases, Lewy body disorder with the ventricle enlargement, supporting the notion that there might be an inflammatory component in the background of Lewy body disease associated ventricle enlargement. And inflammatory doesn't mean that there is an infection. It means that there is a kind of an inflammatory response in the choroid plexus, eventually producing more CSF as a compensation. So this is the summary of our little study. So first of all, the ventricle enlargement correlates with the aging-related tau astrogliopathy, which supports the notion that there is a kind of a traffic jam or barrier dysfunction going on in the background. Second, the subependymal periaqueductal protein pathology seemed to correlate with some of these MRI variables and distinctly in the Lewy body versus the tau-related diseases, which kind of not so far from this involvement and dysfunction of the pulsation and effect on the CSF circulation. It is a very nice observation, and we have to see whether this can be translated to any clinical measure or marker, that as the third ventricle gets wider, that reflects more pathology in the brainstem. Maybe if you can use it as a surrogate marker for follow-up or longitudinal to say that then this patient has more pathology. And finally, this inflammatory component in the choroid plexus only in the Lewy body disease seems to be worth further considerations. Now, in summary, the recent ideas about the ventricle enlargement and cerebrospinal fluid adds a lot of new components like the interstitial fluid, the lymphatic drainage, the vascular components, and even the choroid plexus. So evaluating and the innervation of the ependyma and the function and the ciliary function of the ependyma. So it seems that in a way, these neurodegenerative diseases do have an effect on these. And it seems that maybe there is a distinctive effect. So maybe the Lewy body disease will have a more choroid plexus plus aqueduct component versus the Alzheimer's disease or the amyloid, or they may have a more reflecting an interstitial fluid component. So if we can translate this to the measurement of the ventricle components, it might be helpful to translate it to the predicting the pathology. So the take-home message of my presentation is that ventricle enlargement seems to have something to do with inflammatory alterations in the choroid plexus or alterations in the interstitial fluid G lymphatic system, and the physiological function, pulsation of the CSF circulation might be affected in these neurodegenerative conditions. And this is our team, and I would like to thank Yoon Lee and Ali Karakani for the immunostainings, and all the team for the patience and support that they provide me every day. Thank you very much.
Thank you, Gabor. Any questions from the audience? And let's also have a look at the chat. Okay. Well, interesting data. You showed new data just for us today. Thank you.
One question to Gabor. We have also the concept of PART, primary age-related tauopathy. And principally, as Heiko Braak also presented, lots of this tau pathology starts actually on the level of pons. So can one speculate that maybe we have tau pathology in subependymal region on that level that might explain also our idiopathic normal pressure hydrocephalus cases, because then you will not see anything in the cortex.
I like your idea, Irina.
You don't like it?
I like your idea. I think it's exactly what would support the notion, and that would mean that if we have an idiopathic NPH without any Alzheimer or other pathology, maybe it's only an accumulated aqueduct subependema pathology. That's great, Irina. Thank you. Wonderful.
Thanks. Thank you, Gabor. Wonderful study. In the spirit of the association we're trying to seek between hydrocephalus and degeneration, did you get a sense of any correlation between pathology type, pathology distribution, and degeneration and cell loss?
You mean that generally in the full brain, you mean?
Yeah, well, in the regions that you examined.
Because in this study, I focused only on the subependymal, so I did not correlate with the full brain pathology. I was trying to address this hypothesis whether the subependymal area has any pathology. What you say would be the correlation with the, for example, the Bragg stage or whatever phase, which I did not present in this presentation, but it's obviously a great comment. Thank you.
Anything else? Okay, I have a couple of questions. Actually, I was thinking, first of all, I understand the logic, but are you saying that this pathology is somehow affected the function of the ependyma and therefore the flow itself? So if so, can you do also structural analysis of the cilia? Can you do a microscopic analysis of the ependyma to see how the cilia looks like and correlate that with the presence of pathology that you described?
You know, we can do microscopic analysis, which is, we don't have standardized methods to evaluate this in the neuropathology practice, how the cilia looks like. You can do electron microscopy or certain stainings for the cilia. In this study, obviously, I didn't do that. That would mean additional months of study, but it's a potentially a good idea. The other, what they usually associate with dysfunction of this is so-called little verruca formations in the aqueduct, which can be also measured with or evaluated in our study. But what you say is a distinct study, which is a very good idea and deserves, but it is many months of additional work.
Because putting the, so wearing the hat of a reviewer, you know, if you were to send me this paper, I would like to see a correlation. The more the pathology, the more the impairment of the cilia.
Yes, okay.
That will convince me of the argument. So
thank you.
I guess we'll be the reviewer anyways. But the other question I have is, okay, when do you do this in NPH?
Sorry, when?
When do you do the same type of analysis in NPH?
I mean, I can't do it because we don't have. But what Irina just said. But what Irina said is that if you do the same evaluation even in a few cases, whether the subependymal area have tau pathology, because in elderly individuals, they always have in the locus coeruleus. So potentially that can affect this area, or eventually have the NPH patients have much more there compared to the normal aging.
Yeah, and the comment I want to make in this regard is that sometimes it's difficult to disentangle what's coming from the problem. I mean, what's coming first? I mean, for example, Alberto, there was a time when Alberto thought NPH was a real disease, and he published a case of a man who died, unfortunately, after surgery, and they didn't find any pathology in the brain. So, and actually they say, okay, we thought it was vascular dementia. Actually, there was a time where you thought that vascular dementia also existed. So, it was a misdiagnosis of vascular dementia. Then he got a sternal lumbar drainage. He improved. They went for the shunt. the patient died. They did pathology and didn't find any sign of neurodegeneration. The only thing that they found was what they interpret as a stretch injury of the corpus callosum. They found some ubiquitin positive inclusion in the corpus callosum. Where I'm getting with this is that we've reported only one case of an extremely effective, too effective shunt. The patient did amazing, but we programmed wrong, I don't know. But basically what we saw was a stretch injury coming from the rapid change of the ventricle size, which caused hyperintensity of the corpus callosum. Actually looked like Markafama-Bignani disease. And this made me go back to the paper you published and say, maybe what they saw was just a stretch injury, maybe coming from the hydrocephalus, but also from the intervention, because this man had been, he was treated. And along the same line, I will argue that anything you find in an NPH patient, if you were to finally analyze the area around the aqueduct, it would be difficult to prove that that's the cause, because the aqueduct actually sustained an important stress. The CSF flow in NPH at the aqueduct level is crazy, and actually is used in some studies with MRI to confirm the diagnosis. And we don't really do it because I don't think it's that accurate. But this is to say that there's a huge shear stress going on there, and it's actually been hypothesized that some of the cilia issue that is seen in NPH is actually coming from the stress, from the mechanical stress of the very fast flow. So that's, food for thought for the future when you finally have the chance to look at NPH brains, but that can be another possibility.
These are great thoughts. And let me tell you one thing. There is one thing that I didn't show in this lecture. So we evaluated one more thing, and I would address your topic. I will tell you in the coffee break. I didn't want to, because I show here
which we don't have.
that are not published, and we just finished it last week. But there is one more component that we evaluated, and that would support your thoughts, and I hope that will add a more dramatic twist to the story.
Okay, so stay tuned for the next symposium. What can I say? Okay, so anything else?
To ask something.
Oh yeah. Okay, please.
Maybe not. I don't know.
Oh no, we just see him on the screen. No, okay. Okay, so if there is no other question, we can move on to the fourth and final speaker of the day before the panel session, with Alberto Espei, a dear friend, we're known for, 15 years at this point. And I have known him for so long that I can tell all the times he changed mind and on diseases and everything. So while, are you set up? Okay, good. So Alberto is gonna describe and discuss this idea that neurodegeneration can present as with a ventriculomegaly, but I'm sure, knowing the way he presents and his thought process, that he's gonna give us food for thought, deconstructing NPH maybe. Alberto was the first author of a paper published in Annals of Neurology. I don't know, you're going to talk about it probably, but that paper has been used as an argument for the non-believer of NPH to say that we should even stop doing VPS in our patients until we have a clear idea of what we're talking about. So quite controversial paper. So Alberto, please take it away.
Thank you so much, Alfonso. Thank you so much to all of you for being here and your interest. Also, those of you virtually, I'm just delighted to be with you all. This topic is one that really we all should think about, at least, because we will have patients with this condition. And when I was growing up in the field, this was a reversible cause of dementia. And there was a pathologic diagnosis, which we are not talking about here, but it had to do with the questions raised before about ependymal lining and thickening of the meninges. We're not talking about that. We're lumpers. We neurologists are the lumpiest of all fields of medicine practitioners. And I think that's part of the problem. Every other field of medicine splits, we lump. So I'm gonna go through a little bit of a travel here through the history. This is disclosures that I tell you for sure will not have a relevance here, but I wanna go through a bit of a journey with you, and some of it is related to what Alfonso had already pointed out. This is the list of things that I believe are clear pitfalls in the field. And the most important of them all is the one on top. We're dealing with a diagnosis that's defined by a response to a treatment. That's a problem. That's how we define it. We do an open label assessment of someone's gait. And if we see there is improvement, if we ask objective and subjective measures and there's an improvement, we say, yeah, that's the condition I suspected you all along had. All right, well, it's difficult to diagnose something that's so darn non-specific to begin with. This that you see here is an old slide. You can see the dates and the reference, very old. And it is now much worse because we now have a whole bunch of other things we think are lumped into it. But we think it's a syndrome that's very characteristic and that is still something we're missing. The trouble is that, of course, everything about it is happening with age in a variety of conditions, in a variety of neurodegenerative conditions as well. And here you see examples of two individuals with vastly different outcomes, and they both kind of had a bit of a response to it. The gold standard here, again, is the assessment of gait. You see that the true positive, true negatives, sensitivity, specificity are all really based on that idea. And of course, a lot changes when you're trying to understand what the true gold standard is. So the prediction is, as you see here, all over the place because really what you're essentially uncovering is an outcome to an open label assessment of an intervention. And that's 3 months, but can be sooner. A lot of the literature, as you'll see later, very short duration. And the farther you go out, as you'll see, the less of a response you notice. So a lot of this is short-term outcome. So essentially, the way we think of the diagnosis is that we have somebody with hydrocephalus, and let's say that we go through the process of defining that someone might respond to VPS, and this is usually large volume tap or lumbar drainage. And if there is sustained response, we'd say that's it. Now, again, sustained response is defined differently by different people, but by and large, we say, if that happens, that's a diagnosis. The problem is that you have all this thing in between where something kind of happened, and there are degrees of how much and for how long it happens. And of course, the issue with this is that it is really quite subjective. And it is well known that the placebo response to interventions of the kind that are required to assess response, and as well as the ventricular peritoneal shunt placement, is very high. So this is sort of pretty much the way we do this. We essentially have an open label testing, and we then have an unblinded evaluation of that response. Now we can say, oh no, but we have ways to quantify that with an electronic mat, etc. But then we're dependent on the time. We're dependent on the time. The time will tell us if we were right or not, but we already have made a diagnosis. We have to, right? Because we're putting the patient through a neurosurgical procedure. So this is an example of someone who had a very un-NPH history. In fact, you look at this is a man with a vascular disease. Vascular disease is a vasculopathy. He has a number of vascular risk factors, has had a couple of admissions to the hospital for sudden onset freezing, and look, hypertension, hypercholesterolemia. And if you look at the MRI, you see white matter abnormalities. This is, of course, the classical presentation of vascular Parkinsonism, or so I thought at the time. We succumbed to the pressure that the physician that referred this patient to us had said, no, this is NPH for sure, and we're going to prove it isn't, and we did a lumbar drainage. And of course, this is a couple of months after that. You see how much worse he was on the left, but how much better with a 3-day in-hospital lumbar drainage procedure he got. And so he actually went ahead, and this is in fact the one case that Alfonso was thinking, that I diagnosed as vascular Parkinsonism because it meets criteria. You have just 3 elements for the vascular Parkinsonism criteria. You need Parkinsonism, which you could argue he was low enough to be Parkinsonian. I hope that most of you disagree with that, but that's often the case, and that's why the literature is filled with the Parkinsonism phenotype here. Vascular disease because of imaging abnormalities. And usually we jump into the idea that it's vascular because white matter hyperintensity must be vascular. And then a connection between the two. And that's the third criterion for this Stoltzman criteria of vascular Parkinsonism. So he met that, but on pathology, of course, he had NPH. Meaning nothing else, and just, again, thickening of the ependyma and of the meninges and nothing else. And in fact, the third pathologist, it was before the time of Gabor Kovacs, otherwise I would have involved you in this case. This was many years back, but the third pathologist, actually knowing that I was convinced there must be some vascular disease, said to me that there was an increased density of the microglia in the frontal lobes, indirect evidence of vascular disease. And that was probably a way to shut me up. I think I needed to. Now, we miss something that in the same scan is, this is what we typically like to see. You look upwards and you see that what you saw before is in fact enlarged subarachnoid space. So it's not just the ventricular enlargement in NPH, is the subarachnoid space dilatation. If you have only ventriculomegaly without subarachnoid space enlargement, it's probably going to be something other than normal pressure hydrocephalus. In most cases, you don't see a response, although I'm going to show you examples of that being accepted. But by and large, that's how we think. So this is sort of a way to think through this. We believe that a stepwise decline that has vascular appearance, both in imaging and history, we're likely dealing with the vascular Parkinsonism. And of course, it can all now be within, and it's been well demonstrated, the context of normal pressure hydrocephalus. And it's interesting because look at the literature that we have accumulated. This is titled Vascular Parkinsonism, Clinical Correlates Predicting Motor Improvement After Lumbar Puncture. Here's just the beginning of the story I'm gonna weave through this particular presentation, the lumping. Here we're saying vascular Parkinsonism, but we're gonna shunt these patients because surely there is some degree of hydrocephalus. And what's interesting is that you do find that there is some way of a response in some cases, not unusual if you do a short-term response. And in fact, quote, some patients diagnosed with vascular Parkinsonism improve after lumbar puncture. Non-responders more closely resemble PSP. And look at the vascular disease. When have you seen that any vascular disease would decline in burden by extracting fluid? You would question that if it's vascular or not, right? And all what you saw before, by the way, was what we today refer to as transependymal exudate. But that you can easily make the case because somebody has hypertension, hypercholesterolemia, 60-year smoking history, that must be vascular disease. Well, then it shouldn't really shrink if we do extraction volume. Now, this particular patient is belonging to the third category of pitfalls, which is this idea of biomarker usage. And also the order of symptoms. This man had forgetfulness to begin with. And we think that that's not the right presentation for NPH. We think it's a gait presentation, not a cognitive presentation. And actually, you can see this, the phenotype will be consistent with it, but look, forgetfulness and a tap, that large volume tap, no response, and then biomarkers, as you see here on the right lower corner, that show that everything is low. Low beta 42, low total unphosphorylated tau, which is the pattern that has been well described in dementia with Lewy bodies. This man had a episode of hallucinations, but it turns out that he actually ended up undergoing a shunt elsewhere. This is one patient, I think, that NPH does exist because he actually did have this to hip. I don't know what happened to the video. I don't know what happened, but essentially he responded beautifully to it. This is actually what you see here in the right is 4 years later, I convinced him, because of course he never wanted to see me ever again. I was the doctor that said this was dementia with Lewy bodies, no response to TAP, cognitive onset, biomarker profile consistent with DLB. I was the doctor that had it wrong. And I convinced him that I said, hey, if you come, I'm going to confess to everyone how wrong I was, but also use it as an example of how we could improve in selecting individuals that could do actually better, because all of this is just a success to me. And of course, not to my credit, of course. Now, what I wanted to show you here, and the reason I'm doing this is because it turns out that this is well established that the hydrocephalic condition that we think is associated with NPH actually does alter the drainage of the APP products. And so you have low products across, and that if you then test that again after a shunt, then the levels go up. And so that's important. And that is mostly with A beta products, but not necessarily with tau. Now, I want to show you at the very end something that's just very, very new about this, which I think is fascinating. So it turns out we think that memory deficits should really not be the onset of what you would call NPH. And most studies have shown that cognitive onset do not predict sustained response. And that's important, because if we then see the opposite, where it's actually gait onset and then urinary frequency, then we see that. By the way, I'm showing you an example of enlarged subarachnoid spaces with entrapped sulci. This is the kind of profile that you'd like to see. And in fact, ideally, you will see no memory deficits. Ideally, you don't see the triad, because those patients that do best are people who do not have cognitive impairment established. All right, now moving on to the main area of concentration for this talk is this concept that NPH, or what we call NPH, may in fact be the presenting feature of neurodegenerative diseases. This is one of several examples. This is a man, as you see here, seen with very little interaction with me, Parkinsonian, with retrocolis. And if you hear the term Parkinsonism with retrocolis, what did you think? Think of PSP. Yes, and in fact, examine this man, and you can see that there is here the eye movement he was almost ophthalmoplegic. It has to be PSP. Showing him, of course, because with the hydrocephalus, one of the considerations was NPH. Now, the pre-mortem diagnosis, we were convinced was PSP, but in pathology, he had dementia with Lewy bodies. Dementia with Lewy bodies. That was, the best lessons, as far as I'm concerned, are the ones where I'm wrong, and I've been wrong so many times. I've learned so much. Right, exactly. So what did I overlook? Well, one of the most important things I overlooked was hallucinations. And I just minimized them because the phenotype to me was classical for just one thing and one thing only. And I looked in the literature, it turns out that retrocolis isn't pathognomonic of PSP. It's been demonstrated to occur in DLB. But the other thing is that I missed cognitive fluctuations. I needed to learn that, in fact, there was a time when I've seen the patient. I saw the patient twice, exactly looking like that. And the spouse said to me, yeah, it's interesting that every time we saw you, it's when my husband was in one of those states. So I've been seeing him in the context of a cognitive fluctuation, which now today we know is a reflection of hyperperfusion from paroxysmal orthostatic hypotension, which may not need to be orthostatic, just simply paroxysmal hypotension in patients with cardiovascular dysautonomia, which is a very common comorbidity in DLB. Now, here you have another example of a man that just didn't do very well after a knee surgery. You can see wide-based of the gait, short steps, external rotation of the feet. This is the classical description of PSP, because you see that it's also very normal, relatively normal movement of the upper body with relationship with the lower body, which in the past used to be referred to as lower body Parkinsonism. But he would start falling, which is probably something that shouldn't have happened. However, the pressure was on for us to really consider shunting, and the lumbar drainage, in fact, shows that he really objectively didn't have much of a change, but subjectively this was amazing. He then underwent ventricular peritoneal shunt, and the response was very poor. He died, and his pathology was PSP. So what did we emphasize? Of course, the fact that there was hydrocephalus. What we did not emphasize, and should have, is the fact that even at the very first visit, there was a loss of the optokinetic responses, which is a very sensitive test to define slowness of saccade. Sometimes we cannot tell that, and that will be, of course, inconsistent with NPH. So that's very important. You can see here that the hummingbird should have been our clue if we had paid attention to that, but we did not. We in fact said, yeah, this is probably, and I think that we also wanted to believe, and the field of neurology is based on belief and hope to the detriment of science. So we actually, at that point, we sort of thought, oh, you know, the patient you saw at the last is in the last column here, and these were the 4 patients that had by then come to us with the possibility of NPH. None of them had it. You can see the diagnosis here, not only PSP, but DLB, in some cases with some degree of vascular disease, but very minimal compared to what we would have expected if this were the case. So when you look at this from a historical perspective, you run into articles that talk about this progressive supranuclear palsy with normal pressure hydrocephalus comorbidity. And this is one example. You see how I'm highlighting clues that in this patients thought to have an NPH. In fact, something else was developing. But closer to home, and this is Tony Lang's paper from a while back, looking at the patients that at the time had been seen by him and Terry Curran here at the Western, and you can see that of the series shown here, in fact, these patients with communicating hydrocephalus, what we today would say NPH, perhaps, had very much PSP. Virtually all of them, and some with pathology confirmation. So what's emerging is this concept that NPH either coexists with or increases the risk for something else, and almost makes us think we need to start counseling our patients with NPH that NPH is an unlucky condition. They should expect to have a second condition different from that which we're using as a diagnosis. And here is sort of how this is all presented. coexisting. And part of the problem, of course, is what we're now seeing. Even you would think this is historical, and now we're looking. This is much more recent, and you can see that we're talking about an overlap. We think these are two pathologies that overlap. That they're very frequently common. I would argue that we neurologists make a diagnosis and can never revise it. We say it's NPH, and if something emerges, it's not like that, you know, God, it's interesting. That thing we call NPH must not have been, because look what emerged. We say, this is NPH that evolved into something else, and now you have two pathologies. This is the same thing with essential tremor. This is with everything. We are a field, I have to say it, very self-assured. We think our diagnosis should never be contested. Essential tremor that evolves into Parkinson's is ETPD. Yeah, ETPD is the dual pathology pitfall, because we refuse to think that at point 1 must have been essential tremor, at point 2, Parkinson's appears to be the case. Of course, we're right at point 1. Come on, we're neurologists, we're super smart. And results here, by the way, is that when you look at patients who have PSP and NPH, no difference in terms of neuroimaging features. Neuroimaging features are what we use to diagnose NPH. And we're here trying to put together groups of people with PSP and NPH to find out how they're different, and we find that they aren't. Problematic. And of course, here you see another example. Look at this. Higher Evans index in PSP than MSA and in PD. So, of course, like what you saw before, over a third of patients with PSP meet criteria for NPH. So NPH and PSP, quote, share pathophysiological characteristics, because we cannot say that NPH was an incorrect diagnosis. I think that's problematic. And as a field, we need to start trying to be a little more humble with the idea that if we call it NPH and something else evolves like a hummingbird bee sign, right, that probably wasn't NPH to begin with. But look at this interesting thing. In that particular paper, and this we're talking about a very recent paper, 2022, you see that if you see Parkinsonism with hydrocephalus, imaging supportive of NPH, and even you're saying, gosh, there is no difference with the PSP. It doesn't matter. Do a CSF tap and just go ahead and shunt. And then look for a midbrain atrophy. And if there was something like that, then do a DAT scan and then see what happens. And you probably will see that people with PSP might not have a sustained response. People with tau PET, if you can do them, also will not. But if they don't develop, they may have a better response. That all seems interesting to me as a way of saying NPH and PSP are part of one construct here. So the first paper that actually did this in a long-term scale, time-wise, was this one. This is a series from the Mayo Clinic. They had a 9-year experience. Now, Alfonso referred to this paper, and one criticism was they see few people over that span of time. Another possibility is that they are very, very selective, and that ultimately those patients that go on to have VP shunt placement are very well selected. So the people that actually end up going from 41 to 13, those 13 must have the thing, because you were so selective that, boy, those for sure have NPH. And look what happens. I think this is a study I've learned tremendously from, and it was a very much an eye-opening study for me, because you can see that the sustained response at 3 years was really left in very, very few patients. The outcome data, as you see here, for the 12 patients that there was data, by the 3rd year, only a third had a response, so 4 out of 12. And look at the interesting data from a clinical perspective. all of these patients have postural instability to begin with. So pool test failed. None of them responded by 3 years. That's a feature of PSP. Cognitive impairment, if you had it, only 1 of 8 patients had a response at 3 years. Urinary incontinence, only 1. Only those that had gait difficulties alone were the ones who improved. I think that was very interesting. Look at this is the diagnosis for those that had pathology. Now, remember, this is a really well-defined cohort of people that for sure, because we just very few, right? You are really hyper-selected, the best of the best. So most patients with NPH in the most stringent way defined will not have sustained responsiveness according to this study. So what we did is we did our own cohort. We looked at a 10-year experience. And I wanted to briefly tell you that over a 10-year period, we got 142 cases. And we do all that we do. From the beginning, almost 50 cases did not have NPH. We thought they didn't have it. And then we did either external lumbar drainage or large volume tap to then rule out another 62. Now, this is imperfect because I showed you an example of someone that we said no NPH based on what we see, and we were wrong. So there is this error built into this. But by and large, we think that what we do is predictive to some extent of the response to shunting. So we're down to 3 cases from 142 cases. 3 that went in, 1 added out of the protocol, came directly through our surgeon and bypassed our protocol. So 31, and look at this, at 12 months, 11 cases had lost benefit. At 24 months, another 6 cases. By 36 months, 3 years, 4 cases, another 4 cases. So essentially you're left with 10 cases with improvement by 3 years. And we think we're really good at pre-selecting 2. Well, that comes about to about the same, 1/3 of sustained response by 3 years. And the cases with revised diagnosis at the end, we had pathology for, as shown here. DLB wins with 3, Alzheimer's 2, PSP 2, and vascular dementia 1. So this is very interesting. So really, just 32% retain response, and the revised diagnosis in the cohorts shown here in over 25%. So we may not have all diagnosis, but we know that something else is up, and whatever this is not NPH as much as we'd like it to be. Here's an example. These are just 4 cases to make the case that you have a hard time distinguishing them on an imaging basis, and yet they have vastly different responses. So this concept that somehow there is a specific profile on imaging that predicts how much someone will respond, how long the response will be, is not that easy. It's not that easy. So I think it's important to know this. Now, here's an example though of somebody that did respond very well. And you can see how bad this looks on a superficial manner. And this is because I think what you see here most is not the ventriculomegaly, but the enlarged subarachnoid spaces. And I think that's, very, very important. And this, by the way, look how MoCA of 27. This is somebody that if you just briefly look at this, close your eyes, you say, God, dementia for sure. This is look right. And then you start looking at the pattern doesn't look, this is a pseudoatrophy pattern. So the drop fluid pockets can be confused with atrophy. So these are the patients we really need to look for because they are the ones that would do very well with shunting. For now, though, we just think ventriculomegaly. Let's do fluid diversion to understand how many of them will benefit. And then if there is no response, of course, we think is something else, and it's typically a neurodegenerative condition. And sometimes people do respond, but the response is unsustained. And as you see here, it can be 12 months, 4 months. And so in our case, sustained response defined at 3 years, really only 3% of the shunted cohort and 7% of every NPH diagnosis sent to us. So this is the opposite to what we've been told, that This is a condition we don't recognize enough. We're missing a lot of. This is a condition that we overdiagnose. And I think it exists, but it's rare. And of course, it's important to find it because I am haunted every day by that patient that I show you, right? That I miss. I was convinced it couldn't be NPH. It had to be dementia with Lewy bodies because it fits criteria. Look at the biomarker profile. So we looked at the data on the studies that were done, and essentially, conclusion that can be said is that the longer you wait, the more attrition there is, and studies with a follow-up duration of greater than 2 years had, interestingly, just 1/3 less or 1/3 benefit left at the end of it. So again, the same concept, the longer you follow them, the less of a response you see, and this is significant. So hydrocephalus can be a presenting feature in neurodegenerative diseases. You often think of NPH, and the final diagnosis could very well be something quite different. And I think that when we begin to lump here is when we get into trouble. If we see Parkinsonism, if we see early postural impairment, if we see falls, oculomotor impairment, hallucinations, we really should be thinking not on NPH, regardless of what the ventricular enlargement is. In fact, most likely the ventricular enlargement will be a sign of parenchyma that's becoming smaller, of atrophy. And I think cases of dual pathology are not two separate diseases coexisting, but rather the fact that some of these diseases present with hydrocephalus at a time that we think initially could be, of course, NPH. But look at the kind of studies that now we see. This is also this is a case report of somebody that was diagnosed as PSP, had NPH features, and the pathology was corticobasal degeneration. This is now 1 year shunting. And I have to admit, though, there is a response. So if this is in fact done at 1 year, you say, well, there is a response in 1 year. So this brings me to the last pitfall, which is this idea that I don't know who I am to define sustained response. I think the sustained response definition is in the eye of the beholder, and I would actually say in the eye of the patient, right? Because if you get a patient that responds for 6 months, whatever the underlying condition is, maybe those 6 months are pretty good. And who am I to say, I predicted you will not have sustained response. So I'm highlighting this issue because it really is one that I think we're all gonna struggle. And if in fact there is some degree of disability that comes from hydrocephalus in patients with PSP, maybe we need to still do it. So I'm not suggesting that here's NPH is very rare and here's everything else and is much more common, and let's not try to then go on and do flow diversion. We need to figure out where we're going to continue doing. And I would argue that having a low threshold is okay. And I know this is different to what some have argued in terms of abolishing this. I've never said we need to put a moratorium. This was actually the editor of Annals of Neurology when our paper, the 10-year data, was published in Annals. He said, this is it. We should really put a moratorium on this. And I think, no, that's not really what we just need to know what we're dealing with. And ultimately, if nothing else, to explain to the patients what the correct prognosis is and the uncertainty about the prognosis. So you remember this particular paper, I just showed it to you earlier, and I'm bringing it back because this ethical dilemma is at play here. It says that the diagnosis of PSP should not preclude patients from the choice of shunting for a still transient improvement being reported. And I have to admit that when I first read that, I said, no. Maybe I'm wrong, maybe yes, maybe we could still do it, but who are those individuals that could benefit for some time? And is that some time meaningful? And look at this though, the second part is that we propose midbrain atrophy as a neuroimaging marker in NPH patients, and that is wrong. That's part of the lumping. So yes, we can consider shunting in individuals with ventriculomegaly that we think may have a PSP, but no, midbrain atrophy is not a sign of, we cannot do that. We cannot do that. So I think that we really need to move away from lumping. I hope to have convinced you that the problem we have is ourselves. Is the idea that we have self-imposed the role of arbiters of what is and isn't. And that needs to end. That needs to end. Just because we can put an order to the chaos of the phenotypic diversity doesn't mean that that is the truth. That is just a truth to us, but it might not be anything nature recognizes as such. So let's stop lumping. Let's recognize that what we do is very much unblinded assessments of an intervention, and all interventions have an embedded placebo effect. Let's end the short-termism. It should be termism or just durationism. I think that if we measure success or lack thereof with very short outcomes, we miss both ends. We will have false positive, we'll have false negatives. And this NPH determinism or the dual disease pitfall, that if something strikes us as NPH at 0.1 and something else emerges that questions that, let's question it. Let's not say, well, it's interesting, this patient had two diseases, NPH and PSP. I don't think that's correct. I think that people aren't as unlikely as we think they are, and that NPH is not a condition that increases the risk for a variety of different things. NPH is a syndrome, and I would say it's a placeholder diagnosis for a variety of different things, some of which is what ultimately comes to be PSP. So it doesn't, as you can tell, have a specific clinical, imaging, or even a pathologic hallmark. And it is dependent on the response to an intervention. That's defined variably according to how long we want to follow these patients. No definitive test confirms or refutes the diagnosis of this condition. And so I argue that at the very least, when we say NPH, we think that parenchyma is sick, and that sickness may in fact be a neurodegenerative disease. So as far as the future research, I think that there continues to be equipoise about an interventional treatment that would have never been approved if this were a medication. If this were a medication, we would never had it approved. This is approved because we see people that do very well, but by and large, in how often we use it, this would not have been a condition that we would, or a therapy that we would continue to use. So I suspect that we need to just be mindful that there is a syndrome here. And if in fact we agree there is equipoise still for therapy, we need to test who are the individuals that are actually improving with this, who are those who are harming, because we are harming some people. And the protocols need to be very much widespread, volumetric. We should by now, one of the simplest things to do is volumetric. We're doing it now in all of our patients, but it's amazing how often we still are just doing a very visual inspection, and we say, this enlargement of the ventricles is disproportionate to the parenchymal atrophy, must be NPH. That should not be. We need to understand what is a response, what is it? What's the length of it to qualify as responder? What the kind of nature? Is it gait alone? Is it gait and cognition, etc.? This ethical cutoff, as I just mentioned. And then finally, the role of biomarkers. And I've shown you how tricky that is at the moment. But I want to end with this very recent publication that shows that there is a difference in both proteomics and all other AD biomarkers, depending on whether you do the fluid drainage from the reservoir in the ventricles versus you do them in the lumbar region. And we were thinking, let's not add a layer of complexity, but yes, that's happened. And you can see how different those can be. And this particular paper argues that we cannot directly extrapolate biomarker levels measuring lumbar samples to ventricular CSF without a lumbar cranial index for the molecule. So with that, I wanted to thank you for your attention, and I'm hopeful that we can have a productive discussion. Thank you.
Thank you, Alberto. Is there any question in the audience? Sunil.
It's also more of a comment. Nice to see you again.
Good to see you, Sunil.
So the comment I have is as well related to this expectation bias that we have with loss of response. So I think in your last slide, we have to add a specific definition of what defines loss of response in cases like this, because there's a device. The device is changing its quality over time. Brain compliance is changing with respect to the device as well. So I think there has to be some standardization as to is it true loss of response, disease progression, combination of the two, because we may actually be overestimating the loss of response in certain cases. And I get it. There's no standardization for this in any of the studies I've looked at, but I think it is a factor that plays a role here.
No, absolutely. It is very important to figure a way to standardize the response for sure. It would be so helpful if we could really get this, lacking a biomarker that tells us yes or no, this is or isn't NPH. We are at the mercy of the response, the judgment of response, both to the fluid drainage that happens before someone that we end up thinking is a candidate for shunting, and also after shunting, because you see people that respond in some way and then do not with shunting, as well as the opposite case, which is the one that terrorizes me, as I've mentioned. So that we need to do. And you're raising a point. How much of it is device dependent? Perhaps I haven't thought about that particular end of the response. One thing that we find in the patients that we follow long term with NPH, and I'm using air quotes because the kind of patients that end up phenoconverting into something else. I hate that term, but it's just what we think of him, is that there is an increasing need to essentially lower the drainage and eventually just stop it altogether, because eventually those patients actually do worse if you keep them at the same rate of drainage. But how do we tell how much of it is the actual device and how much of it is the patient's is unclear? I would think it's mostly the patient. I think it's a messy study. People, I'm sorry, can you give the microphone to Neil? Because people are...
I was just saying, and I don't know if the study's already been done, but I've been curious if about repeating a large volume tap in a shunted patient at the time of non-response. And what's the data?
We do it when there is lack of response at some point to rule out dysfunction, and or when people have been treated in the past with other shunts that we cannot adjust. And when we see improvement, which we do the shunt using a new valve so we can adjust and maybe shunt more. So it's mainly done in terms of troubleshooting. We also done in a couple of cases sham lumbar puncture, so that like telling the patient up front, look, we're going to do 2. I think that this idea that there is a placebo response to the tap test is misleading. I don't think there is so much placebo as we think there is, from my experience at least. But I have a lot of things to say, but I'll shut up for now. Yeah, there are.
First of all, I enjoyed your talk quite a lot. It was really, really nice. There are a couple of comments. First of all, this from clinical point of view, when you are talking about 6 months response, 1 year's response, 3 years response. For example, if it happens to be a PSP patient that progresses fast, then 1 year is a long time, which means that that patient benefits from it. So in a sense, I don't feel that this length of the response is so important, rather the response in itself. That's one comment regarding clinics. The other thing is that it was so nice how you presented that something starts as normal pressure hydrocephalus and then evolves into something. So then in the end you have PSP or you have LBD or something else. So it's sort of the initial sign of this disease. Now, then you have those that principally do not evolve into PSP, CBD. But you have to remember that we pathologists, we don't see everything today.
See, most
I mean,
things.
it took, it's only last 10 years that we have known about frontotemporal lobar degeneration and its pathology. Gabor presented very nicely that in the subpial region, you might have these protein aggregates. Maybe they are only there and they are not in other places. Maybe they progress in different speed in different individuals. So I will repeat once again, we need to do the postmortem examinations on these patients. And it's not good to be dogmatic that this will happen or that will happen, but we need to find the answer to what's really there.
Yeah, thank you.
So I enjoyed your talk very much.
I appreciate that. Thank you so much. And I agree with your points, definitely. So thank you.
Anything else also from home? Do you see any comments in the chat?
Oh, yeah, I see comments in the chat too. Well, you get to. Oh,
Thanks, Dr. S. Very nice to see you again.
great to see you.
I have a quick question. So how would you navigate having a conversation with patients, given the prevalence of NPH in neurodegenerative disease, with regards to the intervention of shunting?
It's a great question, and it's a difficult answer because we essentially have to admit uncertainty, and we do the best to try to remove emotions from the testing during what we mostly do is large volume taps. We used to do external lumbar drainage, which has been shown to be more sensitive, but the logistics of doing that with every patient is just very, very complex. So a large volume tap is what we have. And sometimes we've done it twice. So when we think that there's probably not the best day to test, and we just do it again. But by and large, we try to remove emotions because we say, this is a test. We want to be as objective as we can, because we're essentially trying to give ourselves a glimpse into what life would look like with a shunt. And we explain that sometimes we all want it to work so much that we emerge from that particular testing thinking that we really saw something meaningful. And we explained that's why we try to dampen the enthusiasm, not because we don't want that, but because we want to make sure that what we see is objective. And that's why we measure things objectively and so forth. So let's say in the situation of somebody that has a response, if the response is robust, and we rarely see that, where you see 50% improvement in gait velocity, which, by the way, gait velocity is the most important of the parameters predicting response. If we see something like that, we say, God, this is great. Most likely this is going to be benefit, and we hope it's going to be a sustained benefit. We introduce the concept that this is sometimes seen in people who develop other diseases, but by now we don't see anything else. So we think that the risks are worth, in your case, very much, because the benefit are expected to be high. Now, when the response is there, but we are 18% or 25% benefit in gait velocity is good to have, but it's not something that is overwhelming. If there's objective benefits, and then we say we think that there will be a benefit, but we don't know if that benefit will be sustained. And we just want to introduce that concept there to say, if it were to last for 6 months or a year, where you are going to work and feel better, perhaps as good as you felt with this, would that be worth to you? And that's essentially saying you'll have this benefit for that period of time. Would that be worth to you, even if you lose it later? And when we've presented it that way, everybody has said, yeah, I take anything, right? I'll take anything. So we say, all right, well, then let's proceed, right? And so that's how we present it. We just want to make sure patients are informed that there is a component of uncertainty in terms of the long-term response or sustained responses. And so, and then we just see what happens. And we all see people that are doing great years later. So there's this group of people that benefit tremendously from it. It's just that they are not as many as we would like them to be. But did that respond sort of the counseling? Would you introduce something else besides that, or this? No. Okay.
Okay, so there is a question from Dr. Taylor again. So why was there so much, maybe this is for Gabor more than anything, why was there so much ventricular enlargement in CBD? Was there any change in absorption of CSF through the graduation? I don't think we have data on that in CBD. And also CBD is a rare disease, right?
We don't have data.
Yeah, we don't know. So we're going to have our panel discussion now. So it's 5:15. We're perfect on time. I've never seen anything like this organized by an Italian. So much. Okay, so can you please come on stage? We have the speakers plus a few guests. We have additional panelists, is Dr. Carmela Tartaglia and Dr. Dave Tangway. So please have, and Dr. of course, Irina. Sorry, your surname.
Irina Alafuso.
Okay.
Irina.
Okay, thank you. So we're going to have a conversation about what we'll be discussing so far. But while you sit, I want to start going back to what Alberto was saying, because Alberto, please have a seat, because there is a fundamental problem, because you're criticizing the field for being too dogmatic when we say NPH is a disease, blah, 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 and instead these people have other problems. Alberto, I'm talking to you, don't get distracted. But Alberto, you are also dogmatic when you say, no, this is not NPH, this is PSP. As a matter of fact, if you do shunt in PSP patients without ventriculomegaly, or in Alzheimer's disease patients without ventriculomegaly, it's been done, they don't improve. So clearly we're dealing with something a little different than just the disease that they had in origin. So that's something to keep into account. I don't know if you can answer to this one.
No, your comment reminds me of the historical fact that Tony, if he were here, might say, which is that David Marsden never ever diagnosed a single individual with functional dystonia, for psychogenic dystonia as it was used to be called, right? Because prior to his time, every patient was psychogenic. Every patient. There was no such thing as dystonia in the manner we know it today. So he went the other way. I don't think that I came across in that way. I don't think I came across that going all the way. But let's face it, we are in a position where if NPH diagnosis is made, we don't ever question again. And I presented that case to say, you know, all cases really are examples of people evolving from one point where we make a diagnosis X to another point where we make another diagnosis. Let's think that at least a possibility is that the initial diagnosis was incorrect.
Or that we're dealing with a biological process that is more complex than we think.
Correct. No, and that's always going to be true. But the issue is, what about
you had your chance to say what you had to say. That's it.
What about that's what I'm talking about. The labels. The labels.
Yeah. No, I just want to give the opportunity to others to talk. And actually, I want to welcome Carmela and Dave on stage, so to speak. So we talked a lot. You haven't said anything, so please talk for the next half an hour.
Well, thank you, and thanks to all the great speakers. I do think that this is a heterogeneous condition, and I think all diseases that are in the neurodegenerative family are heterogeneous. And so at some point in time, we will become like oncologists, and we will have molecular precision. And so some NPH patients have alpha-synuclein. We're looking at Alzheimer's disease. Some of them have elevated neurofilament light chain, which shows that they are more neurodegenerative or degenerative than others. So I think at the end of the day, by lumping, as you say, I don't think we're going to move forward. I think we really have to become more splitters and become more precise towards all brain diseases, especially neurodegenerative. And I think the problem to date has really been that we haven't spent that much time. We use a lumbar, we do a drain or that's as precise as we get. Usually we just do a lumbar puncture and say, yeah, they've improved by this many seconds or this much. So I think there is an NPH. I think that the molecular biomarkers will help us.
Dave.
No, I agree with Carmela up until this point. So one comment, I think you need a behavioral neurologist with you.
Do you mean for the patients or for him or for
both?
Right.
Because just to be fair, some of the cases you presented were very rapid in force, which is unusual for what we see here. And I like it that you missed a couple of details. Huh?
No, just, he's making his own career out of his mistakes. Looks like. No. But jokes aside, I always make fun of him and say, next, what are you going to deconstruct next? Facial palsy? Because at this point, there's nothing left.
No, I think I've agreed so far. I think there is some NPH. I think we do see really clear cases. And I have one with neuropathology with zero neurodegeneration and had midbrain atrophy. People thought it was PSP. And I said, no, this is different. It's changing different. Its phenotype is different. Did not have the classic eye findings, and it was pure NPH. Yes, on imaging, I think you did the pathology and zero neurodegeneration, absolutely none. So there is this entity that exists. Yes, yeah, no.
Degeneration of the midbrain.
Right, but it was an MR finding. It was not a neuropathology finding. It was an MR finding. So, and this. No, on the opposite, no, complete opposite. The complete, and I think, to be fair, the clinical picture is really important in defining this condition. I can't stress that, how often Alfonso and I get consulted on patients who, based on imaging, is thought to have NPH, but really when you dig down to the details, it's, no, there's something else going on, or in addition, or when it's NPH plus neurodegeneration or core pathology, but I can't stress the history. And you've actually demonstrated it for us too.
I think DASH is not well known. That's the big, DASH helps a lot, but nobody talks about it. So not even radiologists sometimes.
It's also like the comment, it is often misdiagnosed or misinterpreted as cortical atrophy.
Yeah, that's
it is not cortical atrophy.
I would like to have a short question to all of the panel members. So listening to the discussions, can we identify 4 groups? One is neurodegenerative disease without ventricle enlargement. Group 2, neurodegenerative disease with ventricle enlargement not responding to shunt. Ventricle enlargement, neurodegenerative disease responding to shunt. Ventricle enlargement, no neurodegenerative disease, shunt response to shunt. So the question, yes or no, why don't we do more CSF examination in everybody who has ventricle enlargement, even if we suspect neurodegenerative disease? Because if I understand, for example, Irina's comment, even if in a PSP patient, we say that there is a 1-2 months, a little bit better something, isn't it worth to do more CSF testing in neurodegenerative disease with ventricle enlargement? May I have your brief opinion? Yes or no, or it makes no sense?
Yes, I think so, because we got a lot of information, and this is the future. On the last ADPD meeting, we got to know a lot of new markers that we can use for diagnostics.
Because I think that we don't use too often the CSF testing
in Scandinavia. It's very common, both in Finland and Sweden, that you test CSF always. So on all our patients, they have done the AD, the beta and tau. So that's sort of a routine. And I agree that it should be done more. But then you need to combine it with everything. So you combine it with radiology, with the clinical presentation, and I repeat myself, with pathology, because if you don't put together all the pieces, we are just presuming, we are just, guessing, having our hypothesis, but we need to come to the conclusion, because I think there are real NPH who do not have the pathology we know about today. But it is probably a degenerative disease as well, but it might be a completely new protein, or one of our proteins in a location we just don't identify.
Yeah, no, I liked your schema of these 4 groups. I agree with that very much. I also agree with the idea of using CSF for biomarker development. I think that the interpretation of that is tricky now, as you saw with the hydrocephalus group, can be misleading because of issues related to circulation, and you might get misleading readouts of what the profile is. And then I added the fact that it matters whether it's extracted from the lumbar region versus the ventricle. So why should that be? But it is. And so I don't know what that means, but that adds another layer of complexity. I like a future, I think Carmela pointed it out best, which is one where we would say to a patient, gosh, you have something that we call NPH, but have no idea what it means. We would have to do certain biomarkers and different, some of them might be blood. Of course, some of them would be potentially CSF, maybe some pathology tests we could do. Skin biopsies, probably, I don't think we'll do meningeal biopsies or things of the nature. And then we'll know what you have, and then we'll know how to treat you, right? That's sort of, I hope we get to that point. But it seems very distant in the future at the moment. I don't know why it does feel that way, but anyhow, I love to believe that that's actually going to happen.
I do believe it will happen. And I do think that we do need CSF assessments, and we need to be looking for other markers. I deal with another condition of people who've had multiple concussions. And although we focus a lot on chronic traumatic encephalopathy, Gabor has shown that there's many different pathologies, but there are also many people who have no proteinopathy. Now, they have 20 years of severe psychiatric disease, lots of cognitive issues. They have something, we just don't know what it is. And I think maybe in NPH also, we will find, the stretching of the axons maybe has some protein that is being released that we're just not looking for right now. So I like the proteomics study very much. Yeah. And look for inflammation. I think, you've shown there's inflammatory changes. We know it's an element of neurodegenerative disease. Why would it not be an element of NPH?
I actually don't have much more to add because I agree with everyone, but and I know in England they do CSF on every single dementia patient as well. But in North America, that is not occurring. It's just a matter of patient preference. It's availability. It's the ease of doing it. I mean, I personally would love to do it on every single person. That's a Canadian guidelines, no comment. They're changing. Number 6, it'll actually be up there.
May I have one more question to the panel group? Yeah. So, yeah.
No moderating.
Yeah. Just a brief question to everybody, because there is one condition that in Central Europe, it has a history, and also in the Japanese literature. In North America, usually they don't use this, but there is a condition which was defined by 3 imaging and neuropathological definitions. One is the ventricle enlargement, periventricular leukoencephalopathy, which on a pathological level associated with macrophages, so myelin breakdown, so not this transependymal fluid. And a third, very severe basal ganglia lacunar state. This was described by Binswanger and called Binswanger encephalopathy. And there is a literature for this because it turns out that this is not a hypertensive vessel related or diabetes or whatever else, but it seems, and this is completely forgotten, but in the 1980s and 1990s, there was a literature that this is associated with the fibrinogen level in the blood, so a hypofibrinogemia type of disease, which is completely out of the, I've never heard it here, but like in North America, but I did see a patient or 1 or 2 here who did have these classical signs, dementia, and then also NPH-like clinical symptoms, and then they do not respond absolutely for shunt. So my question is that, just briefly, do you ever consider this or not heard about this, but kind of consider this condition ever in your practice?
You probably saw mine. I think I'm one of the few neurologists who still diagnoses Binswanger. It was something I learned in residency. They are slower than NPH in my clinical experience in terms of development. they look like NPH, but there's a different clinical quality about them. And I look at an MR, the ventricles are not as large as I would like them to be for NPH, and there's a lot of white matter changes in them. But I have diagnosed it before. And I'm not that old.
Yeah, I trained in London, Ontario. We talked about Binswanger disease. So I must say I do not make that diagnosis very much. So I, will have to consider it more often.
Yeah, I also grew up with the same diagnostic label. I must admit, didn't know about the hypofibrinogenemia. That's news to me. But it's interesting that we think of it as the paradigmatic vascular.
The problem, because it's not a small vessel disease.
Right,
Yeah.
right. But that in part is because the diagnosis of these kinds of things are imaging based.
Yes.
They're imaging based. So you see white matter, periventricular, deep white matter confluent. We think of vascular. That's how we think.
Actually, there's a question for you in this regard, since you have the microphone now. Is Miguel C.E.T.S is saying, Dr. Spey, in regard to the white matter lesion, because most of the cases you presented had that in common, white matter lesions in the brain, at the brain MRI. The question is about whether you think that there are certain late onset, this is a topic you like, late onset leukodystrophies that could be explaining some of this.
Very good. Thank you, Miguel. That's a good question. Indeed, we have begun a project to look at this. In fact, we've been running it for a number of years, where people who come to us with leukoencephalopathies, and we just use the term leukoencephalopathy, not otherwise specified, for whom we really don't see necessarily a great deal of clinical imaging correlation, for a study that looks into genetic adult onset leukoencephalopathies. And in fact, we had combined this, we've been collaborating with Marcelo Kaufman, a neurogeneticist from Argentina, and we together, Cincinnati and Buenos Aires, put together this large series. And over a 3-year period, we're approaching 20% of patients with a genetic identifiable through whole exome sequencing disorder explaining the leukoencephalopathy. So we're missing these patients. And I think that if you look at the imaging, if you just do a Google search for CSF1R leukoencephalopathy, which is actually
done it yet.
Huh?
We do this all the time.
All the time. CSF, you had your CSF1R encephalopathy clinic. That particular one, if you look at it, it is remarkable how unimpressive the imaging abnormalities are. And for sure we're missing them, because we say, oh, somebody with hypertension, probably that's what it is. And we sort of downplay the role. So to Miguel's point, I think it's important for us to think of adult onset genetic leukoencephalopathies, explaining some examples ostensibly of anything in the spectrum of Binswanger to even very mild small vessel ischemic disease, as our neuroradiologists like to interpret the white matter abnormalities.
Okay, thank you. So,
well, if I go past, have I received any brains with the question Binswanger? No. If there have been some questions regarding white matter disease directly, no. If there have been questions regarding vascular lesions or vascular dementia, that is sometimes coming in, but usually there are very mild pathology regarding vascular, because it has completely changed. When I was younger and worked as a resident, there was lots and lots of vascular pathologies, but then we had the beta blockers and we have the statins and everything, and the brains look better. And probably that's why we live longer, but there is lots of degeneration instead. So, but we are poor in assessing the vascular pathology, undoubtedly. So that needs to be looked at.
How many of the vascular dementia diagnoses you confirmed on autopsy?
Maybe in approximately 45 to 50 cases, only 1 or 2 were correct. So it's very unusual diagnosis.
Yes, we think of it. And
Reading why you think about vasculates, this Hashinsky's stepwise progression rather than anything else. And I think that's not how it goes. So it's overdiagnosis on the vascular because of that.
No, we work at the same department.
Okay, so Carmela, please.
So going into this genetic, I had one who was sent to me for NPH, lots of white matter, and it's a TREM2 mutation. And that's only recently been described. So of course, there'll probably be more of those in the literature.
Okay. Is there any question from the audience? Because we have some questions from the, we address those. Anything from you guys? Okay, so in case, just raise your hand, let us know what you think. I want to switch gear just a little bit. And this question I want to ask is based on the comment you made before when you said that the patient is sent to the cognitive clinic if they're found with beta at the autopsy, sorry, the biopsy, because at the autopsy will be a little late to be referred, a little too late. Yeah.
If you have a patient that comes with normal pressure hydrocephalus, that's the patient of a neurologist and neurosurgeon. Whereas the patient with A beta in his or her brain, that's a patient of geriatrician. And for example, Parkinson's disease, and there have been lots of discussion, patients with Parkinson's disease, they go also to neurologists, but when they have Lewy body disease, they go to geriatrician. And they are, at the moment, they are quarreling who should have this patient group.
And that's what, this is what I was getting at. So this taps into the model of care. Because historically, NPH has been seen as a neurosurgical disease, okay? And the reason we do it here this way is because I had an interest in Gates disorder. That's why I was interested in NPH, but neurosurgeons here didn't really want to do it. And then I say, well, if you take care of the patients after, we'll do it. That's why, as they knows, I program the valves. That's why I'm pretty convinced that we're dealing with Sunday, because I can tell when they get better, and if they worsen and they come back, or add an MRI and the setting of the valve changes. So I see these on a regular basis. And I manage these patients with the mindset of a neurologist that is used to chronicity. So I'm also okay with seeing their worsening over time. This would be like doing saying to a Parkinson's patient, I will not do brain surgery, deep brain stimulation, because in 5 years from now, you might be in a wheelchair because of balance problems. We still do, but that's besides the point. What I'm getting at is that we managed to create this system where it's a collaborative effort, which I guess everybody will agree is the best way, blah, blah, blah. But we also realize that there are challenges and the country is different. So what do you think, talking about a wish list, will be the ideal flow of a patient with this disorder, with NPH? So obviously you come late or you come during the biopsy, but starting from you, where do you see, where would you like this field to go in terms of clinical care, simple clinical care?
First of all, at least in Scandinavia, the patient comes to the, how you call it, the open care system. And there you meet a physician who will listen to your complaints. And then you will be forwarded to the special clinics, which is a neurologist. I think neurologist is the right first point. The problem in our society is that first doctor you meet doesn't always understand that this patient should go to the neurologist. That's one of greatest problems, because then this individual might have these complaints a longer time, and get other complaints on addition. So he or she doesn't always come in time to the neurology clinics. Then when you are in this so-called special clinic, like university hospitals, the neurologists and the neurosurgeons collaborate well. That's the example regarding this in Kuopio, where these two guys were sitting at a dinner and decided that, well, your patients are very much like mine. And when I shunt them, I always have this piece of tissue. So instead of throwing it away, I can send it to you to pathology labs. So the collaboration regarding these two specialties is very good. Geriatricians are subspecialty in Sweden. So we don't have them in, I think there are 2 or 3 of those in Finland. So that's the third person who comes in. And their benefit or their knowledge is in that they really know how to handle the aged subjects who have also often cardiac problems, they have diabetes, they have other problems. So I think these 3 specialties are actually needed. And the main thing is that each one respects each other so that they don't compete with each other, that no, this is my patient, I shouldn't get involved in this, and et cetera, et cetera. And the competition is sometimes a problem. Yeah, and but it's like once I was reading, and this is not in the field of NPH, but it's in the field of medicine in general. I had there was a patient who died in a small hospital, and he was demented. And in my opinion, you always need to do a post-mortem investigation to know what's the cause of dementia. Yeah. However, the son was very concerned because his grandmother had also been demented, and he had read papers and this might be genetic, etc. So he talked with a physician who signed out the death certificate that, well, I would like to have a post-mortem examination. And then this physician told him that, well, why? Well, I would like to know what the disease is. Well, you can't learn it through an autopsy. So it was really, and then finally he called me personally and I called to this physician, and I was furious because he repeated to me as well that I didn't know that you can find the root cause of dementia via an autopsy. So education is tremendously important so that everyone does what is expected. So I think we miss lots of cases regarding postmortem because they go to these smaller hospitals and healthcare systems, and those some of the physicians are not aware that the diagnosis that you have written, NPH or Alzheimer or whatever, that there is a big chance that it's not correct.
Especially if it's on purpose.
It's about the care. What do you think the care of this patient? And I will add to that also the effect of time, because we are not discussing strong enough how relevant is timely diagnosis to these people. We've seen now the damage of COVID because a lot of patients are now not getting the treatment. I saw a patient a few weeks ago, 3 years waiting for us to see, and he was mobile by the time he was referred. From the picture, it looked like NPH. Right now, he was in a wheelchair, demented, and we're talking about doing nothing at this point. It was too late. Do you want to say that?
Actually, it is a big problem in our system too, because those patients had to be identified by the GPs. And actually, most of the subjects that come to the neurological clinics are already cognitively impaired. So this is a problem because then the pathology also is more advanced.
Actually, temporal sequence, I think, true onset of dementia, I had it wrong in that particular patient when I thought cognitive impairment was the onset. That would be a red flag is if it's true. If it's a dementia syndrome, then it's unlikely to be NPH, or at least a condition in which hydrocephalus treatment with the shunt will improve. When I came to Cincinnati, there was an NPH evaluation in the hospital, and people were admitted to the hospital for an NPH evaluation. And essentially, we were just called and get to see these patients. They were in the hospital already. And it was pretty perfunctory of me to come in and say, well, I have some doubts about this. Yeah, sure, large ventricles. Thank you for your consultation. The shunt is scheduled tomorrow. I mean, so it didn't almost matter what I said. It was pretty much, and there was a misadventure, a patient that actually had had a cerebellar stroke, and of course, large ventricles, but that was me. They put the shunt and there was acute decompensation. And at that point I said, we no longer will accept consultations in the hospital for NPH evaluations. Because while I agree that early treatment is desirable, NPH, as we know it, isn't an emergency. So if you have somebody that has to come to the hospital, that's a red flag against diagnosis in my mind.
No, we're not talking about emergency, but we're talking about years.
Right. No, absolutely. Years. Eventually it happens. But to your point, though, we had someone that we were convinced was NPH, but it was very mild. And it was no cognitive difficulties. He was a professor at the university doing really well, but otherwise walking was the main issue. No, even though urinary incontinence, but the imaging was as consistent with it as it was. He didn't want to be, right? He said, no, I don't want to have this done. Well, years passed, we kept on following him, and eventually he fell and had a complication from the fall, actually almost induced a myelopathy. He had a cerebral, cervical injury, and then went into a wheelchair. And it was too late for us to do anything, but he would have been great to have done, as you were saying a little earlier. So the flow is, I think, important for us to emphasize patients before they ever get into the triad, because I think once you have dementia, I think that's the odds become diminished greatly. Yeah. Now, as far as the evaluation, which is probably your question, the more the better. How to do the pathology, that's hard because that has to wait until you've already made the decision to do a shunt, which means that you've done the large volume tap and then decided, yeah, the odds are that it's going to be favorable risk-benefit ratio. Let's proceed with ventricular peritoneal shunting. And then at the shunting process, then you do a biopsy, but you don't have that piece of data
before
up earlier. So that's the issue with that. But of course, now, by the way, with some patients that have unusual phenotypes, especially if there is sleep-related difficulties with otherwise NPH, we also do
Eglon-5?
Yeah, so we do an encephalopathy panel looking for Eglon-5. That's something else that we're now doing pretty much regularly in all our patients.
Have you found any?
No, not
Because
so
anytime
far.
I think about it, I tell myself, don't bother.
Yeah, right. No, I haven't, but I don't want to miss them, right? That's the thing.
So, okay, yeah.
Thanks. Well, I think, like has already been mentioned, I think education has to start with the family doctors. Like here, the big problem is that any patient who's in that age range of NPH, the usual age range, is first of all told that they're aging. So when they or their families come and they say, he's gotten slow, she's gotten different, it's like, well, this is their age. And so I think really we have to change education, not just about NPH, but in all these brain diseases, first and foremost at the family doctor, nurse practitioner level, because by the time they get to us, half these patients have many other pathologies. And you're saying 4 categories, there's probably many more categories, because in the beginning, if you get to these patients, there's probably going to be a lot of them where they just have NPH, but some of them are starting something else, right? Like they got the 30 years of amyloid in their brain, but they're okay. That's not the issue. So I do think education is going to be key to changing outcomes for these patients.
Dave, what's your ideal flow? Is what we do at Toronto West?
I work with you. This is my dream job.
That's it.
Yeah, that's yeah. So I think each individual specialist brings a piece of good information to the assessment. So I look at it very differently as a behavioral neurologist. I'll rely on Dr. Alfonso for the gait assessment. Love to get more biopsies when we do shunt because that does make a difference in how we think. And I think it's the collective. I think that's important. That would be the most ideal. And to do the assessment in a timely fashion, which has, some institutional issues with that. So that's one point. The second point is, much to Carmela and much to what Alberto has said, is about education, but this is not the group that we have to educate. Unfortunately, there are still neurologists out there and neurosurgeons. You don't have the triad. Yeah. It's not NPH. And I'm like, hell it is. We don't want to wait that long. You cannot wait that long. The other aspect we have not touched, but if Mark Hamilton were here, he would actually say, well, the first presentation of NPH is actually balance before gait. And I totally agree with that. That's the pattern that we are seeing here in our patients. By the time the gait changes, you should be thinking of if it responds to a TAP test or an ELD, yeah, you got to shunt this person. It's balance first.
Very good, very good. There is a question from Christian Espinoza, who is thanking us for the presentations and also asking and wondering whether FDG PET can have a role in differentiating co-pathology in NPH. I have my own answer, but what do you guys think? FDG PET.
I know that's a standard, the Mayo Clinic, when I communicate with them, that is part of the standard workup. We've never made it part of the standard workup, but I actually see a lot of interest in it. I think not so much to define what might be a pathology, but just to know whether it's normal or abnormal, regardless of it. And I think that you want a normal FDG PET.
Exactly. So actually, if anything, in NPH is subcortical. There's hypometabolism of the thalamus and basal ganglia. Caudate.
Yeah, I'd say look for other look for PET amyloid or another PET marker, because I could imagine that FDG PET is abnormal in many people who have maybe co-pathology, maybe are on certain medications, maybe have other things. So I would say
depression.
Yeah, depression.
Exactly.
And also the cost. We also need to take into account the cost, especially in the healthcare we work on. Do you have any comment about the FET? Well, maybe you don't order many PET studies for your this again, Christian is asking whether there's a relationship between NPH and specific PSP phenotype, and he's talking especially about the what used to be called primary progressive freezing of gait, which is now PSP PF. So basically the question is whether it's a specific phenotype of PSP. I just worry that Alberto's answer is going to be long, but maybe just this, because we addressed it in a way, the relationship between PSP and NPH. Is it PSP all along? Is it a subtype of a PSP? Is NPH and coincidentally also PSP? So
if I make one point in my lecture is that it is, when we reach a diagnosis of PSP, NPH was a misdiagnosis. Now, is there a group of people with PSP and hydrocephalus, then the hydrocephalus is responsive for a period of time to shunting? Sure, but I wouldn't call it NPH. I just want to make sure the label is not, oh, these are people with two diseases, one is NPH and one is PSP. I think that the odds are, when, in general, if we develop several symptoms, we develop 5 symptoms, we can have 5 diseases, or we can have one that explains all 5. So I want to make sure
I'm glad that you clarified that.
Yeah.
Because you're just thinking up on the name.
You need to remember that PSP is a tauopathy, and Gabor showed that you can have the subepidermal region tau pathology. PSP starts in the basal areas. So in a sense, it might be that normal pressure hydrocephalus is the first sign of PSP.
The hydrocephalus. How about just hydrocephalus?
Yeah, but at the same.
But the point is that we do not know what is the just hydrocephalus. We don't know the pathological substrate for that, whereas we have lots of other diseases that evolve starting with the
Yeah,
sign.
I think we should stay away from disease. That's the thing. These are syndromes. So I'm okay with calling the way you want to call it, because I know it's just a label and it is a syndrome. So now I understand, because I thought that you were saying that NPH is wrong.
You happen to be sophisticated enough to be able to handle labels, syndrome diagnosis, but in the community, if we say this patient had NPH, and then now a point B is PSP, you say, oh, it's interesting. This patient had 2 diagnoses. Yeah. Now you think different. Oh, no, this wasn't a syndrome. And we know that right? So I think that's why I say, let's just call it what we see it. There was hydrocephalus. We thought it was NP hydrocephalus, but it was hydrocephalus. We call it NPH because we had no better way of describing it at that time. Then you get the whole picture of PSP, and maybe you get the pathology of tau, and you say, oh God, no, this is PSP. And the question is it more likely that this was NPH by whatever definition and now is PSP, or is it that we called it NPH and it really wasn't NPH, it was PSP all along? At one time in point, all we saw was the hydrocephalus and the gait abnormality.
Well, communicating hydrocephalus can be the other.
Yeah.
Actually, normal pressure is
exactly. I think it's more honest. We just,
So what
Well, I think I'm waiting for Ivan to come up with a 4 repeat tauopathy marker so that I can know who's got hydrocephalus with PSP, or I would assume that there will be different strains of it, in that.
I have no comment. It's too hot of a potato to say anything.
Sylvia, anything from you? No? Okay, well, that's, I guess the end is 5:56. We're supposed to end now, and just a few more words from Gabor first. Gabor, why don't you give us some concluding remarks? What you're bringing home from today?
We could just call it ventricle enlargement with or without neurodegenerative disease, responding or not to shunt therapy, and do this type of strategy. I think that the future is optimistic, especially if we could add one of these tests. So I think that this meeting helped us to understand that we are on the same page, maybe use different wordings, but at the end of the day, I think that we had the same conclusion. So I think it was a very nice.
Thank you, Gabor. Yes, nicely summarized. And so just a few final words to thank you all for being here. It's been 3 hours without a coffee break. So, it's really dedication to the cause of NPH, and special thanks to the speakers, the local ones from Toronto, Alberto, who flew all the way here from Cincinnati, an hour flight. And from our special guest from North Europe. Next time we're going to do it in your house. Thank you all.